Can do it now. You can do it. We can do it. Continue. हाँ, अभी बच्चों को आवाज तो आ रही है. You can do it. Okay, now मैंने इसको निकाल दिया.
Hello students, am I audible? Yes, I'm audible, I believe.
You are. चलो bye.
Thank you. Say thanks to Mithun. Hello students, am I audible? Okay. Okay, don't worry. This part will be completely trimmed. We are going to start new. Okay. So, hello students. Welcome to Vedantu NEET English. I'm Ms. Gopika, your own biology master teacher. Welcome all of you to Backlog Series, and we are going to cover all the 11th chapters. It's not as such a series, but then it is basically just to make sure that all your 11th portion is covered. Okay. Ready all of you? Yes or no? Perfectly audible. Now no one has a problem, right? Yes. So students, things happen. Okay. This is the thing with technical issues can keep happening. So I will just share it with the students. I think many of them ran away. Yes, 1 second. Okay, done. Yes. Okay. Yes, so today we are going to do body fluids and circulation, and all of you know body fluids and circulation is an important chapter, not just from NEET point of view, because students, you should always remember that your foundation, whatever you prepare for your NEET, that is what you're going to get carried forward in your MBBS, and it is going to be a big, like it's like an ocean, right? Now you're studying it like in one bucket. Okay, body fluids and circulation, you're going to study just a bucket, right? But in MBBS, you're going to study it like an ocean. But this bucket is going to contribute for that ocean because it's going to be the foundation. Okay, so students, don't worry, all the notes, okay, this notes, body fluids and circulation, whatever I'm writing, I'm going to draw the heart, I'm going to write everything beautifully for you. So the link will be in the description box. Even biological classification, the link is in the description box. Just go click on that link, enter your phone number, download the notes. Okay, clear all of you? So the link will be in the description box, so don't even worry. Now chapter weightage, students, last year 3 questions have come. So generally all your human physiology topics are generally in a weightage of 12 marks. Okay, sometimes it can go up to 16 marks depending on NTA's wish. Okay, they want to put an extra question, they will put an extra question. Okay, I hope all of you smash the like button, also subscribe to the channel. We have to make it 200k. Only with your support we can do that. Okay, so do that definitely. So students, generally all the human physiology chapters are in a range of 12 marks to 16 marks, but a whole, okay, a total, that entire thing can go up to 40 marks to 55, 60 marks, okay, entire human physiology. Okay, yes or no? Yes, exactly. Okay, English channel, but all Tamil Pasangala is here. Okay, Yes. So shall we start? So now when we talk about body fluids in circulation, what do you understand and what is this circulation, ma'am? We all have studied in our smaller classes that circulation is required so that I can transport few things, right? What can I transport, students? I can transport gases. Okay, I can transport gases. I can transport nutrients. Yes or no? Okay, these few things I have learned. Also, I can also remove, right? I can do removal of nitrogenous waste. Nitrogenous waste. So, circulation when I talk about removal, transport of gases, transport of elements, transport of nutrients in my body, right? Yes or no? So, this is also same thing, students. When I talk about circulation, this is the only thing I talk about, that okay, transport of gases should happen, transport of nutrients should happen. Okay, these are going to be done by your RBC. They're going to carry oxygen, remove of carbon dioxide. All this we have studied in breathing and exchange. Then we also talk about removal of nitrogenous waste also, which we have studied in excretion. Saturday excretion class is going to happen. All this we know. But who are the main people who are going to do this? Who are the main people who are going to do this? Now, in animal tissues, we have studied about your specialized connective tissue. Under specialized connective tissue, okay, so under connective tissue, all my students studied what? Ma'am, Ladri saw a snake fight, right? So, you spoke about specialized connective tissue. Students, under specialized connective tissue, where is your blood coming? Ma'am, under fluid connective tissue, what is there to ask? So easy question, right? So, under fluid connective tissue, we talk about two main people. Okay, we talk about two main people in this entire thing. Who is that, students? We talk about the blood and the lymph. Blood and lymph. Any doubts so far in this? These two people are going to help with the transport, right? These two people are going to help me with the transport. I have blood and lymph is going to act like the middleman. Now I need to know what is the exact composition of blood. But before that, students, all of you should understand, there are in your lines of your NCERT, it is written that organisms have simple system and complex system. Like in humans, it is more complex. But then you have porifera on the other side, which is very simple. What do? Very simple, ma'am. Nothing they do, right? They basically take in the water. They take it through the ostia. And they bring it out through the osculum, and there is a spongocele in between. They have a spongocele in between. Then, but on the other side, you have humans. I want all of you to answer in the chat box. No talking to each other. Where are you from? Did you eat food? Did you breathe? Did blood circulation happen? No. Just listen to me. This chapter is very important and I'm going to teach you in such a way that everything will seem simple. Oh ma'am, that's all. This chapter was so easy. I'll make you feel that, okay? I'll make you feel that. So all of you back to the class. Who are talking? No, please tell their name. Okay, we'll have class leaders in the class. And yes, did we achieve the target? So we have to achieve the target also. So here, students, we have humans which are more complex. Humans which are more complex. Now, why do I tell they are complex? Because, ma'am, these people have blood, they have blood vessels, they have a pumping organ called the heart. Ma'am, so complex. Okay, then they also have lymphatic system where they have lymph nodes, lymph is there, lymphatic vessels are there. All of this is there, right? So, it is more complex, ma'am. But then on the other side, I have porifera, who is very, very simple. Correct? So, this is a line of your NCERT. I have decoded your NCERT. I have read every line of NCERT thoroughly so that I can just translate it to you in a simpler form. Okay. So, I think the introduction is clear now. What is the circulation and what are we going to do? Now, students, blood components. If I take blood, okay, if I take blood and see, if I do centrifugation, what are the two things I'm going to get? Ma'am, very simple. You're going to get plasma, right? I'm going to get what? I'm going to get plasma. Then with plasma, what am I going to get? Ma'am, you're going to get formed elements. You're going to get, okay, questions also will be asked in the between. So I want all of you to be listening, okay, not do anything else, just listen. Formed elements, or you can say blood corpuscles. These are the components of the blood. Now, what are they exactly, or what is the percentage that they are going to contribute? All this is important, no? So, plasma is going to contribute around 55%, okay, of the total. How much, students? 55%. If this fellow is going to take 55%, how much is that fellow going to take? Ma'am, simple ma'am, 45%. Right? I'm testing your maths. 45%. Okay, 45%. I'm just going to write 45%. Okay. Now they are telling me that there are different things in the plasma that is going to, which is going to play an important role in the future. So what we will do? What we will do? One second. Okay, yes. So, here what is going to happen? This plasma consists of 90 to 92% of water. 90 to 92% of water. Other than that, what do they contain, students? Who is going to tell me in the chat box? They are going to have 6 to 8% of your proteins. And they're definitely going to have nutrients, right? Tell me quickly in the chat box. Very good, Rishita. 6 to 8% proteins will be there. Yes, ma'am. They are also going to have nutrients. They're also going to have some nutrients. All this they have, okay? Now you have to learn the different types of proteins that are going to be there, okay? And their function also you should know. Okay, so the first protein that they are going to have is albumin. My students have heard albumin in excretion chapter when I taught you BCOP pressure, right? Blood colloidal osmotic pressure. That means this fellow is going to mainly help in what? It's going to help in osmotic pressure. Okay, osmotic pressure. Albumin is going to help with the osmotic pressure. Second fellow we have is fibrinogen, and I can see Sukanya has already put the answer in the chat box. What is it? Ma'am, these people are going to act as a clotting factor, right? They are going to help in the clotting. So they're going to help in the clotting, you can write. Okay, help with clotting. Okay, I'll write it. You just need this notes. You do not need any other notes. Every lines of NCERT will be on this notes. Okay? Yes. Yes, very good. Very good. Yes, clotting factor. Okay, help in clotting. Then whom do we have? Okay, we have albumin, we have fibrinogen. Then one person is going to help us with the defense mechanism, which is going to be globulin. All of you have heard about immunoglobulin, right? Ma'am, immunoglobulin I know. IgA, IgE, IgG, immunoglobulin. That means they are going to help with what? Ma'am, they are going to help with the defense mechanism. Defense mechanism. Done, done, done. Done, done, done. Yes. Perfect. Very good. Very good. Okay. Yes. Perfect or not? Defense mechanism. Correct. Correct. Now, when we talk about the nutrients that are going to be present, right, in the plasma, they are telling that some nutrients will be present. Okay. So, this nutrient, students, I'm going to write it this way. Okay. I'm going to write it here. Okay. They are telling that there is going to be presence of some nutrients. What are the nutrients that is mentioned in your NCERT? They are telling that some ions. Okay. Let's say ions. Okay. Ions which are going to be present. That is going to be sodium is there. Okay. Calcium is going to be there. Calcium ion is there. HCO3. Magnesium is going to be there. Cl minus. Magnesium. All these ions they have mentioned. Other than that, they have told a very specific statement in your NCERT that your glucose, amino acid, lipids, fatty acids are going to transit in our body. Let's take example of Charanya. Charanya is in the class. Charanya has had rice. Okay, I had burger. Okay, Meghana has had aloo paratha. Now all of our body will have different amount of starch, different amount of glucose, different amount of fatty acids. Now Meghana has had, let's say, paratha with lot of ghee or butter or whatever. Now she'll have more fat in her body. In the starting, definitely fat is going to be high. As you go forward, it will get digested, right? So depending on what we have consumed that day, what we have consumed, our levels is going to be different. Let's say I had a lot of ice cream, my glucose or sugar intake would be high. So here also they have mentioned a statement telling that your glucose, okay, glucose, amino acids and your lipids, etc., okay, lipids, etc., will be in transit. Okay, transit. Correct? Yes or no? Is it mentioned in your NCERT? Yes, ma'am, it is mentioned in my NCERT. So, students, plasma is going to contribute 55% of the blood. In that, 90 to 92% is going to be water. 6 to 8% is going to be protein. Which are the proteins that you're going to remember? Fibrinogen. So, FAG. Okay, FAG, you can remember FAG. Fibrinogen, albumin and globulin is going to be there. Okay, FAG is better. Okay, FAG is better. Rest everything does not make sense. In that, albumin is going to help with the osmotic pressure. Fibrinogen is going to help with the clotting because this will be in an inactivated stage. Then it will become to an active stage called fibrin. Correct? Then you have globulin, which is going to help with the defense mechanism. Then we have some nutrients and ions will be there, which are going to be your calcium, magnesium, sodium, HCO3, Cl-. All these will be there. With that, what you will have? We'll have glucose, amino acid, lipids, fatty acids, which are going to be in transit. Thumbs up in the chat box. Yes or no if you understood all of this. If you understood, tell me. Now, in the plasma, they have given one last line in your NCERT paragraph that is talking about something called as plasma, okay, plasma without clotting factor. Plasma without clotting factor is called as serum. Okay? Serum. This is what they have mentioned. Yes or no? Last line. Plasma without clotting factor is called as serum. So that is mentioned, highlighted in your NCERT. So make a note. That means without the clotting factor. It could have happened that when a wound has happened, after the reabsorption, the clotting factor is not there. Serum, we call plasma without clotting factor as serum. Okay? Clear? Not clear? Yes? Okay. 47 AB. Does that look funny? Not at all, okay? It's not funny. Yes. Now, next is going to be formed elements. Now, tell me which are the formed elements. You know, 45% is going to be contributed by the formed elements, or you can call it as the blood corpuscles. And which are those? Ma'am, definitely I know. RBC, which is the red blood cells. You have WBC, which is the white blood cells. And lastly, you have the platelets. Okay, lastly, you have the platelets. Let me see how many of you are going to stay till the end of the session, answer all the questions. Let's see that. Okay, let's see that. So, blood is clear. Two components are there, plasma and formed elements. 55% is this, 45% is that. That is going to be red blood cells, white blood cells and platelets. Okay, red blood cells, white blood cells and platelets. Done. Moving forward. Okay, moving forward. Now we will start with your, okay, yes. So students, formed elements, they have some more names, no? So these formed elements, what are they? Ma'am, first one is going to be, okay, first one is going to be red blood cells, okay, red blood cells. I can call red blood cells also as erythrocytes. I can call red blood cells also as erythrocytes. I can call white blood cells. White blood cells as leukocytes. Okay, leukocytes. Okay, K also you can put. Okay, C and K both is allowed. Yes, very good. Okay, then you have platelets. Platelets, which are called as what? Thrombocyte. Thrombocyte. Students, which are also known as, in your NCERT, it is given just below that, cell fragments. Cell fragments. Correct? No doubts in this. Just looking at the spelling, okay, I mean the name. I can say that this is called red blood cells, obviously because of the red pigment. Okay, because of the red pigment. And what is that pigment called as? Ma'am, it is called as hemoglobin. Hemoglobin, which we studied detailedly in your breathing and exchange, right? Detail we have studied in breathing and exchange. Hemoglobin is going to be there. These hemoglobin will have Fe2+, which are going to take the oxygen and they're going to act like the vehicle, right? Like the car or bus which is going to carry oxygen. One hemoglobin can carry how many oxygen molecules? All of you in the chat box, tell me, one hemoglobin can carry how many oxygen molecules? Yes, iron containing pigment, very good. Hemoglobin, it can carry how many oxygen molecules? 4 oxygen molecules, right? Very good. 4 oxygen molecules. So, one hemoglobin can carry up to 4 O2. Okay, 4 O2. Okay. Now, when I look at the name of white blood cells, ma'am, I understand that they do not have a pigment. So, no hemoglobin. Okay, no hemoglobin. Understood. Then these people are called as platelets, thrombocytes or cell fragments. That means these people are not an actual cell. Okay, these people are not actual cells. Not actual cells. Fine. I have basically drew a flag. Okay, orange, green and very good, very good. Okay, orange, green and white. Okay, one blue is missing. Blue also if I put, national flag is done. Okay, done. Okay, now we will learn more about their number and other things. Okay, now RBC, all of you, many of you have already put it in the chat box. Very good. The number is going to be 5 to 5.5 million mm cube. Right, million mm cube. Correct? 5.5. You cannot see it. 1 second, students. I cannot write in straight line. 5.5 million cube. Okay. What about platelets? Platelets, okay, WBC. 6,000 to 8,000 mm cube. Platelets, 1.5 lakhs to 3. Okay. 3 lakhs mm cube. Correct? This is the number. Okay, very, very good. This is the number what we have. Then students, when we talk about the structure or shape, we know RBC is biconcave. In most of the organism, RBC is going to be biconcave. We have also studied in smaller classes dumbbell shape, right? So this is going to be biconcave. Are they nucleated? Definitely in the starting they were nucleated, but later when they matured they became enucleated. Okay, or devoid of nucleus. Okay, enucleated, or we can say devoid of nucleus. Okay, devoid of nucleus. Correct. On the other hand, these people are nucleated, but what is their shape? They are more asymmetrical or amoeboid shaped. Okay, we can say asymmetrical or amoeboid shape. Are you all answering? Come on, tell me in the chats. Very good, very good. Enucleated, asymmetrical, amoeboid. Okay, what is the target now? Yes, students, very bad. Okay, target hasn't been reached. Come on, quickly, let's make the target. Yes, someone put the target in the chat box first. Okay, very good. Amoeboid, asymmetrical, these people are nucleated, okay? These people are nucleated. Now, platelets on the other hand, students, they are not actual cells. Why? Why am I saying so? Because they are actually, okay, broken from a megakaryocyte, okay, they get broken from a megakaryocyte to smaller cells, okay, smaller cells like this. So this is a megakaryocyte. These are a megakaryocyte. These people get fragmented, okay, these people get fragmented. Fragmentation happens, and because of their fragmentation, we get small structures called as your platelets, okay, small structures called as your platelets. Can you see? Yes, very good, very good. Okay, yes, specialized cells which are present in the bone marrow, okay, present in the bone marrow. Even these people are not actual cells, they will not have nucleus, okay. Now, what are the other properties of WBCs that you can think of? Ma'am, I remember that these people have a lifespan, okay. These people have a lifespan of how many days, students? 120 days, okay, 120 days. And where are they formed? They are formed in the bone marrow, okay, formed in the bone marrow. And where do they go and end their life? Come on, tell me in the chat box. Where do they go and end their life? Yes, good job. Okay, they go and their graveyard. Their graveyard is going to be the spleen. Their graveyard is going to be the spleen. Done, done, done. Very good. I can see some of your answers. Now, they are formed in the bone marrow, but in the fetus, okay, in the embryonic stage, it will be in the yolk sac. Okay, in the yolk sac. So that you can just remember it. Okay, yolk sac, I'm not writing it here. Formed in the bone marrow, graveyard is going to be spleen. Yes, very good. Spleen. Any doubts in this? Yes. Now in excretion chapter, I have spoke to you about erythropoietin protein, which are released by your JG cells. They help in erythropoiesis. That means help in the formation of new blood cells. Okay. So the protein that is helping them is going to be called as erythropoietin protein. Okay, which will help in erythropoiesis. Erythropoiesis. Okay, I've written it here. Very good. Now, moving forward, what do you know about WBCs? What do you know about WBCs? Ma'am, WBC students' lifespan is around 20 to 25 days, but in your NCERT, they have just mentioned they are short-lived. So, I am also writing the same thing. They are short-lived. Why these people are amoeboid or asymmetrical is because of their ability to do diapedesis. Okay, diapedesis. That means what? They will be able to squeeze out through the gaps of the capillaries to go and help the tissues. That, oh, tissues have been affected with something, these people are squeezed out. That's why these people are amoeboid in shape, asymmetrical in shape, and even their nucleus also is different shapes. Okay, nucleus also is different shape. Come on, tell me, answer in the chat box. What do you know about your WBCs? Yes. Short-lived, very good. Yes. Now, students, one more thing here. Lifespan. Yes, one more thing here we will add on here, that is around 12 to 16 grams, right, of hemoglobin will be present in a healthy person in 100 ml of blood. Okay, just make a note of that. Okay, I'm writing it here. 12 to 16 gram. 12 to 16 gram in every 100 ml. Okay, one second. Of HB. Okay, in 100 ml, right? Okay, short-lived. Yes, ma'am, these people are short-lived. WBCs are of two types, right? These people have two types. What are the two types that they have? Ma'am, they have granulocyte and agranulocyte, right? They have granulocyte and agranulocyte, which we will be doing in detail again. I'm just going to write it here. They have granulocyte and agranulocyte. Okay, agranulocyte. Okay, done. Now platelets. Platelets also, their lifespan is around 15 days, 10 to 15 days. But these people are not actual cells, and main function of these cells is going to be, they are going to help in clotting. Okay, they are going to help in clotting. And the third and the 13th factor, important, factor number, okay, factor number 3 and 13 is going to be secreted by the platelets, rest by the liver. Okay, secreted by the platelets. Fine. Yes, done. Okay, this is your RBC, WBC and platelets. I have just given a general idea. So if you want to okay, did we miss something? No, right? We'll come to that. Okay, we'll come to that. We'll come to that. Okay, if we have missed something, we will write it here. But yes, here. Okay, here. WBCs. So students, as I told you, WBCs is also called as white blood cells, and they do this thing called as diapedesis. Okay, very, very important. So this is your capillaries. Okay, I'm just drawing like a blood vessel types. Okay, this is your capillaries. Now in the capillaries, there are going to be small pores. Okay, on the capillaries, they are going to be small pores. Now, through this pores, what is going to happen? Your WBCs, okay, your WBCs notice that, oh my God, there is a danger. Okay, WBCs notice that there is a danger. Now, what they are going to do? What they are going to do in the next step? They are going to squeeze out of the capillaries. This question has come in the previous year. Okay, okay, I'm drawing a small pore. These people are going to squeeze out. Okay, squeeze out of the capillaries, go to the affected tissue and help the tissue. Okay, go to the affected tissue and help the tissue. So this process that they do, okay, this squeezing out through the capillaries is called as diapedesis. Diapedesis. Any doubt in this? Tell me. Yes. Endonuclease, if you type in this spelling, they will not smash the like button, they will unlike and go. Okay, please at least put English. Okay, so these are small pores of the capillaries. Okay, capillaries and diapedesis. Squeezing out of WBC. Okay, squeezing out of WBC. Why are they doing this? Because there is a cell that is, or a tissue that they have is being affected. So to help that, these people will be doing this. Now students, most important thing about your WBCs, two types. Okay, WBCs, two types. What is the two types I told you just now? Ma'am, it is going to be called, okay, granulocyte and agranulocyte. Okay, granulocyte and agranulocyte. Now let's draw one structure here. And let's draw one structure here. See, definitely WBCs are irregular in shape, but I'm just drawing it here to make you understand. Okay, don't be like, oh my God, ma'am, you've drawn circle. So, we have a bean-shaped structure. Okay, and here, okay, yes. So, what I have drawn here? What is the difference you notice in my diagram? Ma'am, very simple. You have drawn one which does not have any granules. So this will be the agranulocyte. Agranulocyte. Now you need to remember which are the agranulocytes, which are the granulocytes. Here you can see the granules. These people are granulocytes. Okay, granulocytes. Okay, ma'am. Now, which are the agranulocytes, students? The agranulocytes are monocyte, okay, monocyte and lymphocyte. 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 Monocyte, lymphocyte. Yes? Yes. Now, granulocyte is going to be neutrophil, neutrophils, Okay, I'll write it in order. Eosinophils, okay, and basophils. Okay, eosinophils, basophils. Okay, students, you can join my Telegram as well, Biology by Gopika, where you will get all the notes as well as links of my class. Okay, yes, neutrophils, eosinophils, basophils. Now, there is a trick which I got to know very recently. Okay, that there is a trick that everyone uses. So, I have done a little modification to that trick to remember. The question will be asked on the number. Okay, the percentage of how much these people are going to be present. I think one student already put the the trick in the chat box, but Gopika ma'am has to make new tricks. Okay, so I'm going to write it here. There's no place here. No, yeah. The trick to remember who is going to be in, depending on the percentage. Okay, is going to be never let Mohan. Okay, never let Mohan eat bread. Okay, the trick is never let monkeys eat banana. That also you can remember. This also you can remember. This I modified it. Okay, so never let Mohan eat bread. Now, what is this never let Mohan eat bread? This is going to be in the order of the percentage. Okay, first you have who? N. That means here you have neutrophils, which is around, I'll write the percentage. Okay, 60 to 65%. Okay, then you have L, that is lymphocytes, 20 to 25%. Okay, then you have Mohan's M, which is going to be 6 to 8%. Monocytes, okay, you have eosinophils, which are going to be 2 to 3%. Then you have B, basophils, 0.5 to 1%. So, in the order of percentage, all of you put the chat, all of you type never let Mohan eat bread in the chat box. Quickly, all of you in the chat box, put the trick. Then only you are going to learn. Okay, then only you people are going to learn. Never let M, say you have a friend also you can put. Okay, M, say you can put anything. Never let Meghana eat bread. Okay, never let monkey eat banana. Whichever you want, you can remember. Most of the teachers use this trick. Okay, this is not my trick. So, never let Mohan eat bread is my trick. Okay, so 60 to 65% neutrophil, L for lymphocyte 20 to 25%, M for monocyte 6 to 8%, and E for eosinophils 2 to 3%, B for basophil 0.5 to 1%. Okay, yes, good job, all of you. Yes, Sony, Mohan only we can remember. Okay, yes. Never let Mohan eat bread. So, this is the order of percentage. Okay, now we have to study a little more about granulocyte and agranulocytes. Okay, this is totally on the basis of presence and absence of granules. Okay, of granules. Done? Okay, last time also we had done this box. We are going to do the same box here. So, students, percentage. Okay, eosinophils, basophils and neutrophils. Eosinophils is going to be 2 to 3%. Sorry. Yeah, 2 to 3%. Okay, basophils is going to be 0.5 to 1%. And neutrophils is going to be 60 to 65%. So, this is going to be the largest according to the percentage. Okay. Now, appearance wise, as I told you, I'm not going to draw the irregular shape. Then it will be difficult for me to show the diagrams. So, I will just draw the circles. Okay, I'll just draw the circles. Okay. Now, students, remember this way, they will ask you about the nucleus. Okay, how's the nucleus? So, eosinophils, ear. Think about E for eosinophils, E for ear. On the ear, what do you wear? Headphones, right? You wear headphones. So, here the nucleus is going to be a headphone shaped nucleus. Okay, here the nucleus is going to be like an headphone shaped nucleus, right? Doesn't it look like a headphones? Yes, ma'am, it definitely looks like a headphones. So, here, because these people are granulocytes, we will draw some granules. Okay, headphone shaped, E for ear, E for headphone. Done? Yes or no in the chats? What is Elevin telling? He's marketing. She's marketing for which channel? Okay, done. Very good, very good. Okay, now what is their function? Okay, what is their function? So this is like earphones shaped. Function is going to be, students, they are going to mainly involve in your allergic reaction. Okay, allergic reaction. Main job is going to be involving in the allergic reaction. Correct? Now, eosinophils, basically we tell the names because of their staining. Okay, if they are stained by acid, if they are stained by base, if they are neutral, like neutrophils. Okay, so here eosinophils, here it is stained by acidic dye, because basophils means base, right? Acid, base and neutral. So here also they are going to be stained by acid dye. So we tell that. If you want, you can make a note of it. Otherwise, if you forget it also, it's fine, because it's not mentioned in your NCERT, but we cannot tell. Okay, most of acidic dye. Done? Yes, done. Okay, basophils. Now students, basophils, very importantly, what you have to remember about basophils, here think of it, okay, here these people are going to have an S-shaped, okay, S-shaped nucleus, S-shaped nucleus, okay, S-shaped nucleus. So basophils, S also you can remember. Very, very importantly, these people are the least in number, right? Never let Mohan eat bread. So B for basophils, the least number, 0.5 to 1%. Okay, 0.5 to 1%. Correct? Yes. Correct or not? Yes, ma'am. Perfectly right. Okay, this will be stained by your basic dye. That's why the name is basophils. Okay. Now, these people are going to help with inflammation, students. Okay, inflammations. Inflammations. Okay, these people are going to help with inflammation, again stained by basic dyes. Now, neutrophils on the other hand, okay, neutrophils on the other hand, multiple lobes they are. They are going to be multi-lobed. Okay, so you can draw it. Actually, these people stretch. Okay, these people stretch. Why? Because they have to do diapedesis. They have to squeeze out. So for the stretching, these people will have multi-lobes. Okay, you can draw it like this, okay, like this. Something like this, you can draw multi-lobed. That is very important. Multi-lobe is important for you to remember. Okay, multi-lobed. Multi-lobed. Fine? Done? Yes, S-shaped. Okay, these people are S-shaped, and the students, neutrophils, the main function is going to be neutrophils and monocytes are going to help with phagocytosis. Okay, neutrophils are going to help with phagocytosis. Neutrophils and monocyte, very, very important, helping with phagocytosis. What is phagocytosis? Engulfing. Okay, engulfing of the bacteria. No, so same thing. Phagocytosis is going to be done by monocyte. Okay, this box, okay, can be drawn by anyone. There is nothing like, you know, only this person have to draw it. Anyone can draw it, right? Just the structure. Okay, this is just the structure. Same thing here also we are going to do. Same thing here also we are going to do. Monocyte is going to be 6 to 8%. Okay, lymphocytes are going to be 20 to 25%. Lymphocytes are going to be 20 to 25%. Now again, circle. Okay, circle here also. Oh. Yes. Now students, here monocyte is going to be bean shaped. Okay, what are they going to be? Bean shaped. Okay, bean shape, but then no granules here. Very importantly, no granules here. Lymphocytes are irregularly shaped, or we can just draw a circle. Okay, a circle shape. So this is your monocyte. Again, as I told you, function, these people are going to be phagocytosis. Okay, phagocytosis. On the other side, you have lymphocytes who are going to mainly help with your cell-mediated immunity, right? Why, ma'am, cell-mediated immunity? Because these people are going to be the B cells and the T cells. So, B cells and T cells. Okay, in that T cells are mainly going to be associated with your cell-mediated immunity. B cells are going to be associated with humoral immunity. Humoral immunity. Correct? Done, done, done. Okay, done, done, done. Yes, correct. Perfect. Yes. Now, students, one very important thing about one person, we have to, I can write it in this box. Okay, I'll write it in this box, or we will write it separately. So, basophils release three things. It is mentioned in your NCERT, right? Basophils release three things. What are they going to release? Okay, I don't know if we will be able to write all of it here. Let's try. Okay. I think it's going to be difficult to write it here, students. Okay, I will just write the names here. We will write the explanation there. Okay, it is going to release heparin. Okay, serotonin, and what? Histamine. What are they going to release? Heparin, serotonin, and histamine. Okay, we will just write the names here. Explanation, we will write it in another box. Okay, another box. So, students, this is the structure. Just look at the structure. Okay, same diagrams I have drawn, similar ones. Okay, you can see nucleus is there, neutrophils. Okay, these people are multi-lobed. As I told you, these people are multi-lobed. And remember, their nucleus is like this so that they can do diapedesis. Okay, they can do diapedesis. Now, here also you can see nucleus. Here, what are they? They are like headphone shaped. Okay, these people are headphone shaped. Bilobed, headphone shaped. Okay, E for ears, E for headphones. These people are basophils, where their shape is not like, you know, a little, we cannot like put it under headphones or this type, but then we usually tell these people are S-shaped. Okay, S-shaped nucleus they have. Correct? Done, done, done. This is how the real structure is. Okay, real structure is. Okay, formation nothing, dear, formation nothing. We just have to know why these names have come. Okay, neutrophils is just stained by the dye. Okay, we will not write about the formation anyways here also. Okay, here we will just not use the word there. Just don't get confused. Okay, just write it. Okay, here also I'm not going to add this because some of you are they stick to NCERT a lot. So we will erase this formation word. Okay. Formation means we just tell the staining. Okay, we speak about the staining. Got it? Done. Now, students, for basophil, okay, basophils, we will write it. Shall I write it here? Okay, I'll write it here. I will just erase this. I'll erase this and we will just write it about the basophil. Just 1-2 lines. Okay, 1 or 2 lines. Yes. Okay, what did I tell you? Basophils are going to, okay, basophils are going to release 3 things. Okay, release 3 things. What are the 3 things? Ma'am, first one. Are you all understanding? Are you all understanding? Yes. Hi, Kanjus. Okay. Daisy, no doubt, no? Okay. Basophil, students. First one I told you, serotonin. Oh, board has started dancing. Yeah. Serotonin, okay. Heparin and histamine. Okay, histamine. Now, students, what is going to happen here? Remember that this fellow, who is that? Serotonin. He is a vasoconstrictor. What is he? A vasoconstrictor. So that means, let's say this is your blood vessel. Now this blood vessel can become more narrower, right? Constriction can happen. This is called as vasoconstriction. Okay, this is called as vasoconstriction. So this acts like a vasoconstriction. So because of that, in a small space, more blood will try to go. That is when BP will be more. We have studied this in excretion, no? Same thing like that. When small blood vessels are there and when the blood is trying to move, okay, because of the pressure, BP will be high. Okay, so here this is called as serotonin will do vasoconstriction. Vasoconstriction. Okay, done. Next is going to be heparin, which is going to act like an anticoagulant. Anticoagulant. That means what? Does not allow coagulation to happen, anticoagulant. Okay, whatever lumps are there, it will try to move and it will help the movement to happen. This is why we use this during the dialysis. Okay, yes, thank you Satish. Perfect. Okay, now histamine. Histamine is going to act as a vasodilator. That is why this fellow is going to help with inflammation. I told that basophil function is going to be helping with inflammation. Now, how do you know that you have an allergy? Only if you start seeing redness. So, here vasodilation, students, what is going to happen? Same thing. Here blood vessels are there. Okay, blood vessels are there. These people, okay, the blood vessel is already too big. So, this is the blood vessel. This will go for dilation. That means it will become a little more bigger. Okay, like this will happen. Okay, little dilation will happen. So this will allow more blood to flow. Here BP will be little less. Okay, comparatively to here, it is going to be little less. This will help us to see the redness and the inflammation. Okay, redness. Okay, redness or inflammation. Got it? Done. Any doubts so far? Okay, we have done RBC, we have done WBC, we understood which are the different types of WBC, that is granulocyte, agranulocyte, everything we have understood, right? Yes or no? Perfect. Okay, now we are going to go for what? Okay, so there is a box here, you can see. Okay, your leukocytes, granulocyte, eosinophils, basophils, neutrophils. A granulocyte you have 2 to 3%, 0.5 to 1%, 60 to 65%. Never let Mohan eat bread. Neutrophils is going to be the first, 60 to 65%. Lymphocytes 20 to 25%. Mohan, M for monocyte, which is going to be 6 to 8%. And then you have eosinophils, then you have basophils. Okay, yes, okay, good. Done. Okay, now students, platelets, as I told you, platelets are not real cells, not true cells, right? That is what mentioned in your NCERT. Yes ma'am, these people are not true cells, they are going to be pieces of your megakaryocyte, right? These are going to be, this is your megakaryocyte cell. Okay, this is going to be the cell. This cell will undergo what? Ma'am, this cell will undergo fragmentation. Okay, like this fragmentation can happen. This will lead to the formation of smaller portions. Okay, this will lead to the smaller cells called as your platelets. Okay, smaller cells called your platelets. They tell it is plate-like. Okay, shape is plate-like. They tell platelets, from the word itself you understand, plate-like. Okay, plate-like. Okay, yes, done. Okay, now students, as I told you, these megakaryocytes, okay, megakaryocytes, where are they formed? They are also formed in the bone marrow. Okay, they are specialized cells present in the bone marrow. Just let me know if all the missing letters that is going from here is, coming back here. Specialized cells present in the bone marrow. Otherwise, your notes will look like that Tare Zameen Par, no? All the letters will be flying because the board is taking some letters and going, I don't know if it is bringing back. Okay, so if it is missing, please let me know. Got it? Specialized cells called the bone marrow. Fine, done. Okay, now as I told you, platelets, the 3 and the 13th factor is going to be secreted by the platelets, and these people are mainly going to help with the clotting. These people are mainly going to help with the clotting. Yes, perfect, clotting. And this clotting, students, remember, it's a cascade reaction. Okay, cascade reaction. That means what? One after the other, just like a waterfall. All of you have seen a waterfall, no? Well, how does waterfall happen? Ma'am, what a question, ma'am. How waterfall happen? Okay, waterfall happens like this. Okay, like this. cascade of reaction. That means only if one step happen, the next step will happen. From here to here to here to here. Okay. Yes, this is how the whole thing happens. Now students, in every single step, you will require calcium ions. That is why factor 4 is actually ionized calcium. If you check the entire list, you will notice that factor 4 is ionized calcium, because calcium is required for this cascade reaction to happen smoothly. So clotting, cascade reaction. What is exactly clotting? A wound has happened. Suddenly an injury or a trauma has happened to you. That will be, you will notice that the blood will not flow for a longer time, right? If it's a minimum injury, then the blood will stop immediately. What will you see? You will start seeing a brown, like a lump, okay, brown color, like a clot you will start seeing. What is that exactly? That is because your platelets have got activated. They have started releasing factor, but all other factors will be released by the liver. Okay, will be released by the liver. Got it? Yes. Okay. Okay, students, in case the platelets count reduces, then the blood clotting chances, that is going to reduce. Okay. Okay. Yes. So the clotting, in case platelets, okay, in case platelets count is less, then there are chances that your blood clotting will reduce and you will have loss of blood. Okay, one line is given in there, so you can just make a note of it. Okay, if the platelets count decreases, okay, decreases. If the platelets count decreases, then there is high chance of blood loss. High chance of blood loss. That's it. Okay, one line is given, we will just write that line. Okay. Yes, now the next topic is blood grouping. So have we missed something? Just check NCERT, all of you. Have we missed something in so far? No? Yes? Okay, fine, fine, good. Yes, cytopenia is there, erythrocytopenia is there. We tell cytopenia when there is a less count, okay? So like that, many terminologies are there. Now students, two types of blood grouping you have. You have ABA blood grouping, okay? ABO blood grouping, and you have RH blood grouping, okay? Two blood groupings are there. ABO blood grouping. ABO blood grouping is given by Landsteiner. Okay. And you have one more blood grouping, which is your RH blood grouping. RH blood grouping, which is given by Landsteiner plus Wiener. These two people have given RH blood grouping, okay? Yes. Karl Landsteiner and Wiener, he gives the RH blood grouping. Now, what is this ABO blood grouping, students? Definitely this topic has more in depth. I'm not going into the depth. What is given in your NCERT, we will stick to that, okay? ABO blood grouping means what? Simple things they have told in NCERT, that they will have two types of antigen. Now, what is an antigen? A chemical that can induce the chemical that can activate our immune system. Antigen means what? A foreign substance that is coming into our body. Then we have two types of antibodies which are released by your immune system. I told you, they are like bullets released by your immune system to fight that antigen. So, two types of antigens are going to be there. Okay, so students, if we have to generally, like, in our school also we used to learn like this. Let's say this is your blood groups, okay, two blood groups are there. These are the two blood groups. They just say that, okay, they just draw shapes. Basically, they say that, okay, let's draw this one of the, antigen like this, okay, antigen like this. This is your A antigen, okay, and we will draw one more in some other shape, okay, like this. We can draw box also like this, one more shape. So, these two antigens are there. Two types of antigens are going to be there. So, they told that two antigens are going to be present. Yes, ma'am. What are antigens? They have written in your NCERT. They have told that those are chemicals that can cause a reaction or that can activate our immune system, right? They are the ones, once they enter only our immune system will get activated, right? Yes or no? Why is the energy so less? You people know? No energy, nothing. Then we have two antibodies. We have two antibodies. Which are that antibodies going to be? Ma'am, anti-A, and we are going to get anti-B. These are the two antibodies which are going to be present. What are they? They are from the immune system. They are going to attack the antigens, right? These people are from the immune systems. These are the two things, okay? Finally, yes. Anti-A, anti-B. Very good. Drink water. Don't forget. One second. Yes, done. Okay, that induce response we can write two antibodies, anti-A, anti-B, which are like the bullets, okay, which are sent by our immune system. Okay, sent by our immune system. Now, depending on this, they made a blood grouping. Okay, they made a blood grouping. Understand, there is not only this ABO blood grouping and RH blood grouping, there are many other blood groupings. Okay, but you are only studying ABO and RH blood grouping, which is common among all of us, right? Now, when I talk about RH blood grouping, students, you have to understand few things. This is similar, okay? This is similar as rhesus monkey, okay? Similar as in rhesus monkey. That is why this blood grouping has the same name. Now, what do they mean by this RH blood grouping? Very simple thing, that your blood group can be A positive or A negative. So, this positive and negative indicate the RH blood grouping. 80% of the people do have RH group. Only 20% of the people do not have RH group. Understood? When RH is present, it is going to be positive. When RH is not present, it is going to be negative, right? Okay. Yes. Conjuice. Drink water only after liking, it seems. Very nice. Okay, this is fine. Now, students, this one is going to be antigen A. This is going to be antigen B. Okay. This is blood group A. This is blood group B. This is the blood grouping. I have just drawn two shapes. That's it. I have just drawn two shapes. We used to study like this in college. Okay, I still remember my teacher drawing such shapes. Okay, now we have a box given in NCERT. So instead of that box, I have taken another image to just make you understand how this is going to look like. Okay, so yeah, see how I have drawn triangle. No, they have just drawn that on the outer surface. Okay, so here also we can do the same thing. We can just draw one circle. Okay, we can draw like this one circle like this. Okay, here also same thing. You can just draw a circle like this. This will make you understand the concept better way. Okay, you can just understand the concept in a better way like this. This is the two antigens. Okay, fine. Moving forward. Now look here. This is very important for you to remember. So look here. You have group A. In group A, what is going to be there? Antigen A is going to be there. If antigen A is going to be there, what should be opposite of that antibody? B should be there, right? Because antigen and antibody have to be against. Okay, so here you have antigen A. If we put antibody A, then both will not match, no? So we will put antibody B, so that this tells that you cannot take the B blood group inside your body. Then there is going to be problem, right? RH incompatibility plus your blood grouping. Okay, incompatibility is going to happen. So we will put anti-B. So here antigen A. Fine. Now here what is going to be there? Ma'am, here antigen B is going to be there. So obviously opposite of that antibody A is going to be there. Okay. Now on AB, since both are there, A and B is there, these people are going to have both the antigen but no antibody. Remember like this, AB antibody. AB antibody. So A B is not present. That means antibody is not present. Okay, antibody is not present. So they will have both the antigen. If both the antigens are present, I cannot give any antibody. So here no antibody at all. Here what has happened, students? No antigens. No A, no B. That's why this fellow is a universal donor. He can give it to anyone. No tension, right? So here he will have both the antibody A and antibody B. Antibody A, antibody B. Perfect. Yes or no? Okay, now we have to learn few things. Okay, we have to learn few things. What are the few things that we have to learn? Who is going to be universal donor? Who is going to be whom? Right? How are they giving to each other? All this we have to learn. Okay, now students, let's say here you have A. Okay, you have B, you have AB, and you have O. These are the people that are there. Let's say here also I write A, B, AB, and O. Now who can give it to whom? First, let's start with A. A can give it to whom, students? A can give it to whom? This A fellow, let's say this A fellow, okay, has this antigen. Correct? I'm drawing the antigen only. Okay, I'm drawing the antigen only. Okay, yes. So here what is the scene? Very good, very good. So ma'am, here A can give to A, but it can also give it to AB. Why? Because the antigens are there, right? Here no antibodies are there, so no tension to take. Okay, fine. Done, done. Now, next if you go to B, B can give it to B and it can also give it to AB. Perfect. It can also give it to AB. Okay, I'm just drawing arrow marks. Okay, normal arrow marks, that's it. Now, AB students, AB can only give it to AB. AB can only give it to AB. Correct? Yes, very good. Now O, what about O? O is like, I can give it to anyone, okay? It can give it to A, it can give it to B, it can give it to AB, it can give it to O. Correct? So, from this what do I understand? This fellow, this O is a universal donor. This fellow is a universal donor. And AB, see, AB is getting from orange, green, blue and white. Again, I have drawn national flag, students. I don't know why. I am very patriarchal, I feel. Okay, universal recipient. This AB can take it from anyone. Main reason for this is what, students? Universal donor, okay, the main reason for this is because your universal donor has anti-A and anti-B. Okay, but no antigen. But has no antigen. This is the reason. Then you have on the other side your universal recipient. This person has what? No antibodies. No antibodies. But both antigen A, B, both are present. Correct? This is the reason for them to be universal donor and universal recipient. Okay? Yes, okay. I have been drawing flag, national flag all over. Okay. Now, RH positive and RH negative students. So, this is clear, no? Nothing much is there about ABO blood grouping. Only this much is there. Okay. Now, students, remember your RH positive and RH negative. What is going to happen? A person who is RH positive, that means they have the RH grouping. Okay. A person who does not have that will be RH negative. Okay. Person will be having no RH grouping. Correct? Perfect. Perfect. Now, this can lead to, okay, this condition can lead to something called as RH incompatibility. Incompatibility. Ma'am, how is this possible? Where is it seen? There is a condition called as erythroblastosis fetalis. Erythroblastosis fetalis. That means it happens in fetus, and it is where the RBCs are going to get destructed, right? Yes, mismatching. Very good. Erythroblastosis fetalis. Okay, it's going to happen in fetus, and it is where the RBCs are going to get destructed. So students, here also, if anywhere mixing happen, okay, anywhere mismatch happen, it can lead to clumping. Okay, remember that it can lead to clumping if anywhere mismatch happens. Let's say A is given to B. Okay, A and this is B. Okay, let's say I give A to B blood group. Then what is going to happen? This will lead to something called as clumping. Clumping, which is in simple words called destruction of RBC. Destruction of RBC. Destruction of RBC. Got it? Yes, very good. Erythroblastosis fetalis. This is seen in fetus. Okay. This is happening. So this is the condition. Okay. What has happened, students? The father is what? Is Rh positive. Father is Rh positive. Mother is Rh negative. Okay. These two got, now they have got pregnant. Okay. They have got pregnant. Because of that, what has happened? The fetus blood group is Rh positive. Now in the first pregnancy, generally there will not be any complication until the baby's blood has come in contact with the mother during the pregnancy, during the trimesters. If that has happened, then there is a chance that the baby is in trouble. Otherwise, during the first pregnancy, most of the time problem will not be there, but where the problem starts, during the delivery time, placenta is going to protect. Yes, ma'am, placenta is going to protect Ig foreign substance. During this time, what is going to happen? So, during the first delivery, okay, during the first delivery, the blood, okay, let's say the fetal blood will come in contact, okay, come in contact with the mother's, okay, mother's blood during delivery, only during delivery. Otherwise, the placenta will do the job of separating it properly, okay, during delivery. Now, if this happens, okay, if this happens, then what is going to happen? Okay, others don't have any issues, right? Yeah, very good. So, yeah, very good. So, the issue is going to be, students, the mother's body will start making antibodies. Will start making antibodies against Rh positive. Now, what do you think complication is going to happen? Complication is going to happen. Okay, lagging is there? Antibodies. I did not talk, students. It was not lagging, I believe. I just did not talk. Okay, let me check where the lagging is. Oh, yeah. Now it's fine, huh? One or the other issue is there. Okay, one or the other issue is there. Now it's fine? Okay. So, mother's body is going to make antibodies against Rh positive. Now, the problem lies when the mother is going to have a second baby. The mother is going to have a second baby, then the moment the fetus is Rh positive, okay, if the fetus is again Rh positive, what is going to happen? The mother's body already have recognized this as a foreign substance, and it is going to destruct the RBC. So the second child can end up having jaundice, anemia, fatal condition, the baby will die also, okay. So in the second delivery, so when I turn this side, I will not talk, okay, because I think when I am doing such kind of movements only it is happening. Okay, so during the second delivery, the fetus might have jaundice or anemia. Now, ma'am, do they have a solution for this? Yes, students, definitely solution is there. So look at this. The first situation has happened. So here what we will do, we know that RH, like against RH positive, the mother's body has already made antibodies. Okay, so what we can do is we can use an injection called as Rogam. Okay, Rogam vaccine. Okay, Rogam vaccine injections will be given around after 28 days of the delivery and on the 28 weeks, something on this numbers. Okay, I don't know the exact numbers. So Rogam vaccine will be given, injections will be given to the mother, okay, just after the delivery and during the pregnancy time also, if the mother is RH negative, okay, RH negative. So this is called as RH incompatibility or RH mismatching. Got it? Yes or no? Yes, you can also call this as anti-RH as mentioned in your NCERT. Okay, anti-RH. This is mentioned in your NCERT. So this also you can write. That is actually they're talking about Rogam vaccine only. Okay, Rogam vaccine. Perfect. Done. No doubts, right? Yes, 28 weeks into pregnancy and 72 hours after pregnancy. Okay, 28 weeks into the pregnancy and 72 hours after the pregnancy. Okay, so that is when you can give this. I'll write it here. Okay, and 72 hours after pregnancy. They mean after delivery. Okay, perfect. Yes, perfect. Okay, this is erythroblastosis fetalis. Okay, erythroblastosis fetalis. Okay, very good. Our speed is very good. We have completed so much. Now we will go to coagulation of the blood. Okay, students, actually I try to make a trick. Okay, so because many students don't know who will come first, no, so I made a very funny trick. I mean, I don't know if it's a trick yet. Okay, so you people tell me if you can remember this. Okay, after writing the trick I will show you what do I mean. Okay, see, P PK went to PT field. All of you know PK, no? PK movies, PK. Okay, PK went to PT field. This much you remember. I'll tell you the trick. Can you remember? Is it too difficult? PK, okay, your PK, Amir Khan, no? PK went to PT field. This is the trick. Okay, now we will write coagulation. So students, as I told you, coagulation is a cascade of reaction. Okay, cascade of reaction means like waterfall, right? Cascade reaction, one after the other. One after the other only. One inactivated will make another one activated. It will continue like that. Okay, it's a cascade of reaction. Cascade of reaction. Yes, perfect. Now moving forward. Yes. Now students, first fellow that is going to happen. First, what has happened? One injury has happened or a trauma. Okay, injury or a trauma has happened. That means you fell somewhere, an injury has happened to you. Okay, after that injury, okay, after that injury, what has happened? The platelets got activated. They are like, oh my god, injury has happened, I have to do something. Okay, so platelets got activated. Platelets have got activated, and the first thing that is going to be released is going to be thromboplastin. 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 Thromboplastin got released. Yes, ma'am, thromboplastin got released. After thromboplastin, thromboplastin will activate whom? Thrombokinase. Thrombokinase. Thrombokinase intake will activate whom? Prothrombin. Okay, prothrombin. Prothrombin. Now prothrombin will activate thrombin. Now students, in your NCERT there is a mentioning that thrombin is an enzyme. Okay, thrombin is an enzyme. So in case a question comes, which is the enzyme, you're going to write thrombin as the answer. Okay, because we teach you in a step. So sometimes we will not tell that it's an enzyme. Okay, so when you read NCERT, obviously you will go through that, but still you will write thrombin is an enzyme which is going to activate whom? Fibrinogen. Okay, it's going to activate fibrinogen. Fibrinogen, someone tells, some people tell. Fibrinogen, some people tell. So I am in that some people. Okay. Then finally fibrin. Finally fibrin. Active state. This is going to allow the mesh-like formation. Okay, fibrin is going to allow for what? Mesh-like formation. Correct or wrong? Right? Mesh-like formation will be done by your fibrin. Okay, so soluble to this will become insoluble. Okay, insoluble. This will end up forming the mesh. Now, what do I mean by mesh? Okay, let's say something like this has happened. Okay, this is the wound area. In this wounded area, okay, in this wounded area, a mesh formation will happen. Like this. Okay, mesh formation will happen. And this mesh formation will lead to the blocking of all the RBCs. Okay, we will come to the calcium part, don't worry. Okay, all the blood vessels, everything will be blocked inside this so that we do not have any more blood loss. Now, students, as I told you, this is a cascade of reaction. Every step will activate another step, right? So, in every single place, remember that you will need the presence of calcium ions. Okay, you will take the help of who? Calcium ions in every single step. That is the reason why calcium ion is the fourth factor. Okay, calcium ion is the fourth factor. Fine, I'm going to write it here. So here factor, okay, 4. Students, there is no factor 6, okay. Remember that also, there is no factor 6. Factor 4 is going to be ionized calcium. Oh, where are all these letters going? Are they landing somewhere? Okay, so ionized calcium. Yeah, one has gone and landed there. Okay, calcium ions are required for this to happen. Okay, ionized calcium. Now, students, other than ionized calcium, in your 10th standard, you would have read about vitamin K. Vitamin K also is actually helpful. Okay, vitamin K also is going to help with this cascade of reaction. Okay, vitamin K also is going to help for this cascade of reaction. Students, 1 star has gone missing. Please let me know if it has landed somewhere. Now the trick. Let's come back to the trick. Okay, PK went to PT field. So P stands for thromboplastin. Okay, thromboplastin. K stands for thrombokinase. Okay, thrombokinase. Right, Ven2 has nothing to do with this. Okay, Ven2 has nothing to do with this. 1 second, students. Yeah, calcium is the activating one, dear. Okay, they are the one which will be able to activate fibrin. Now, can you ask me the question, ma'am, why only magnesium, why only calcium in muscle contraction? Like that only, okay? They have to activate, right? Voice is fine only, no? Okay. Now, students, look here. P for plastin, thromboplastin, thrombokinase went to PT. So, here what is that? Prothrombin. Okay, prothrombin. Prothrombin, this is going to be thrombin. and F for fibrinogen. Okay, fibrinogen. And then fibrin. Okay, this is the trick. PK went to PT field. Okay, this will help you understand which order you have to write. Or else you can just write PK PT. Okay, but that will not make sense. So PK went to the PT field will make the entire sense. So P for thromboplastin, thrombokinase, prothrombin, thrombin, and fibrinogen, fibrin. This is a cascade of reaction, how clotting is going to happen. Every single step will require calcium ions, and this is going to ultimately make the insoluble mesh, which is going to help in trapping our RBCs and other thing. During this only, students, when reabsorption happens, serum formation also will happen where the clotting factor is not there. Okay, and this fibrinogen is from the plasma. Got it? Yes. Yes. So, PT March. No, PK went for March. PK went to PT field. Done? Oh. Yes. Now, students, in blood bank, remember they will use sodium oxalate to remove the calcium ion so that coagulation does not happen. Okay, so they will make sure to remove the calcium ions, because calcium ions is there, then this cascade of reaction will happen. So in blood banks they will avoid this. Okay, in blood banks, oh, the star came back. This board is so skilled. The yellow color star I drew, it made it into green star. What a skill. Okay, what a skill. Okay, sodium oxalate will be used so that calcium ions will not exist. Okay, perfect. Coagulation done. Okay, so this is what we tell, cascade. Okay, one after the other, after the other, after the other. Okay, one after the other. Yes. Now, students, here I have just written all the factors. So here you can see factor 5 is there, but factor 6 is not there. Okay, factor 6 is not there. So exactly 12 factors are there, but the names are 1 to 13. Okay, so here you can see 1 only is fibrinogen, prothrombin. See, calcium ions. Okay, calcium ions is factor 4. Students, factor 6 is not there because they tell that this factors, what is that? The soluble, insoluble formally is factor 6. So that's why that is not there. Okay, so yes, and I told you factor 13 and factor 3. Okay, these two are released by the platelets. The rest is done by the liver. Okay, liver helps in coagulation as well. Okay, rest everything secreted by the liver. Okay, how much are we on the target? Now we have lymph, right? We have lymph. Okay, now students, what is lymph actually? As the blood passes through the capillaries in the tissue, some water, very important, some water along with small water soluble substance move out into spaces between the cells, leaving the larger proteins and most of the formed elements. Okay, most of the formed elements. No, Bhavana, you don't have to remember the factors. You just have to remember the coagulation and the order of it. Okay, don't have to remember the factors. Now, students, so what happens? Some fluid will come out. Okay, will squeeze out of the capillaries when it's moving from the arteries to the veins. So how does this happen? It's going to go like this, no? Like this. Okay, like this, right? So when the blood is moving like this, some of it, okay, will squeeze out from the capillaries, and that can be water, small soluble substances, but they will not have, okay, they'll have nutrients. What will be missing? What will be missing? Tell me. Yes, it is a colorless fluid. So the blood formed elements will be missing and larger proteins will be missing. But this can also be called as interstitial fluid or tissue fluid. Okay, interstitial fluid or tissue fluid. So look here, between the cells of the tissue, leaving larger proteins. Whom are they leaving? I'll mark it with a different color. Okay, leaving larger proteins and most of the formed elements in blood vessels. Okay, most of the formed elements in blood vessels. The fluid released out is called as the interstitial fluid. Okay, interstitial fluid or the tissue fluid. Done? Clear? The interstitial fluid or tissue fluid. It has the same mineral distribution as that in the plasma. That means what? 90 to 92%. That means water is there, nutrients are there, proteins are there, formed elements are there, ions are there. All this is going to be present. It has the same mineral distribution as in plasma. Exchange of nutrients, gases, etc. between the blood and the cells always occur through this fluid. That means here lymph is acting as a middleman. Okay, lymph is acting as what? A middleman. Middleman. What is a middleman, students? Now NEET is there, you are there, I am like a middleman, middle woman. Okay, that means I'm helping you learn the chapters, I'm helping you reach your goal, right? So I become the middleman, right? I'm helping you between your goal and you. So like that. So here also they are going to do the exchange. Okay, these people are going to help in the exchange. An elaborate network of vessels. See, elaborate network of vessels called as the lymphatic system collects this fluid and drains it back to the major veins. Okay, the fluid present in the lymphatic system is called as the lymph. Simple, nothing. Okay, whatever is squeezing out from the capillaries, that we call it as the lymph, they will go back to the major veins. Now, lymph is a colorless fluid containing specialized lymphocytes which are responsible for the immune response of the body. Lymph is also an important carrier for nutrients, hormones, etc. Now, one important line is that fats are absorbed through limbs. Okay, one second, students. Baswa sir. Okay, students, an important live will be happening at 7 p.m. So during that time, I'll give you a break. Okay, but all of you should come back. Okay, if you don't come back, then yes, look here. So here they are telling fats are absorbed through lymph in lacteals present in the interstitial villi. So here I am going to show you what do they mean by that. Okay, so students, all of you know how your villi are going to be present, right? So this is how your villi are going to be present. Okay, this is your villi. Correct? Now, if I take one villi and I draw a diagrammatic representation of it, it will have something like this. Inside this, okay, there will be many networks also. Okay, like this, some networks will be there. Okay. The line once again. Fats are absorbed through limbs in the lacteals present in the interstitial villi. That means this is your villi. Yes, ma'am, this is my villi. If I take an enlarged structure of this villi, you will notice that this villi has what? They have lacteals. Okay, they have lacteals. Basically, I have drawn like this one villi and I have drawn the lacteal. So this portion is the lacteal. Because they absorb the fat, it will look like slightly whitish in color. Okay, done? Yes. So students, here you can see interstitial fluid, and if it once it enters the lymphatic vessel, it becomes the lymph. What is going to be there? Water is going to be there, dissolved substance is going to be there, WBC is going to be there, colorless it is, right? But there is no formed elements, that is your RBCs, platelets, all that will not be there. But instead of that, who is going to be there? Formed elements will not be there. At the same time, you will not have larger proteins also. Larger proteins also will not be present. Okay. Yes. So I will make you do some questions once you come back. Okay. Once you come back, I have kept some questions ready for you. Okay. Let's do the question. Okay. I think 3 minutes you people can easily do these questions. Okay. Tell me the answer for this. Which enzyme is responsible for the conversion of inactive fibrinogen to fibrin? Okay. Inactive fibrinogen to fibrin, 2021 question. Quickly you can answer. 7 o'clock. 2 more minutes we have. Yes. And quickly smash the like button. Don't forget to like the video. If you like the video only, they will give you more NEET related content on YouTube. And also many will know that, okay, ma'am is teaching, I have to come and join. Come on, answer fast, fast, fast. Otherwise you can only do one question. Which enzyme is responsible for the conversion of inactive fibrinogen to fibrin? Epinephrine, thrombokinase, thrombin, renin. Yes, perfect, perfect. Okay, very good. The answer is going to be thrombin. Right, answer is going to be thrombin. So now all of you have got +4 marks. Okay, +4 marks in your wallet. Good job. Next question. Person with AB blood group are called universal recipient. This is due to the presence of antibodies anti-A and anti-B on RBCs, absence of antibodies anti-A and anti-B in the plasma, absence of antigen A and B on the surface of RBCs, absence of antigen A and B in the plasma. Okay, what is the answer for this? A person with AB blood group is considered as a universal recipient. Why? Why? You know antigens are present on top of the RBCs. Okay, inside what is going to be there in the plasma? Yes, very good. Naveen Raj, Rani, perfect. Okay, Sony, very good. It is going to be absence of antibodies, that is anti-A and anti-B in the plasma. So this plasma word, RBC word, please make a note. So +8 marks all of you have got in this session right now. Okay, yes. Next question. Match the following columns and select the correct option. Eosinophils, basophils, neutrophils, lymphocytes. Eosinophils, basophils, neutrophils, lymphocytes. Yes, you can tell me the answer. You can do the match the following, okay? Eosinophils, basophils, neutrophils, lymphocytes. 2020 question. Okay, it's 7. Okay, do this question, then we can stop. Okay, then I'll give you a break. Sorry, not stop. I'll give you a break. Okay, this 2 more questions you have. We will come back and do that. Okay, we will come back and do that. Jaldi, jaldi. A, B. Oh, wait, let me tell. Okay, basophils, whatever you know, you please answer. Okay, see, neutrophils, phagocytosis, C2. Two options have C2. Okay, lymphocytes, immune response. Okay, D1. So it is option number C, no? A3, correct? Yes. C. Yes, very good. C, 3 is the option. Okay, only one person told right. 3 is the option. Okay, so only one person gets +12, others get only +8. Perfect. Okay, we will come back and do 2 questions. Okay, heart. We'll be left with heart. Okay, until the live gets over, I can't take the class. So, yes. Okay. See you. Don't go anywhere. Come back for the class, okay? Yes. Ready, students? Okay, last question. Do this question from these topics. Ready, all of you? Should I tell them in the chats? Hello. Wait, let me just inform them. 1 second. Yeah. Okay. Yes, informed. Very nice. Very nice. Welcome back. Yes. Okay, answer this question. Very nice. Roshni, Madhumati, and Ratis, Meghna. Very good. What's the answer? Fibrinogen is going to be blood clotting, globulin, defense mechanism, albumin, osmotic balance. 2018 question. Okay, what is the option? Fibrinogen A2. Option number D. So plus 16 now. Okay, in the chats. Plus 16. How many of you got plus 16? Please drop it in the chat box. I want to see the numbers. Okay, those who got plus 16, those who got plus 12 also drop it in the chat box. Okay, now we are going to start to the main topic. Again, everything is main topic, students. Heart. Right. So first we will just learn about the location. I know you people know a lot about heart. Right, ma'am, it is clenched fist. You know, this much only my heart is, and I have so much problems. All of this you people keep telling your friends and all. My heart is hurting. Okay. So that we will see. First of all, where is this heart located? It is present in the mediastinum. Okay. Between both of your lungs. Okay, between both the lungs. Where exactly? On the cardiac notch. On the cardiac notch. Correct? I've taught you cardiac notch. Yes or no? And students, if you look at the heart, it is slightly triangular, right? And it has moved towards the lungs because it has to be present in the cardiac notch. So it is slightly triangular in shape. Triangular in shape. And we call this portion as the apex. Okay, this portion as the apex and this portion as the base. Okay, don't get it confused. It is because how we have the triangle, no? Like that only we learn this. Okay, like this is a triangle. If you place it like this, this becomes the apex, this becomes the base, no? Similar to that. Okay, so this becomes the apex. And now we have 4 chambers, right? Our heart is 4-chambered heart. 4-chambered heart. And the heart is covered by a double-layered membrane. Heart is covered by a double-layered membrane. What is it called? Called the pericardium. Okay, called the pericardium. Pericardium. And this pericardium will have, again, we have layers to the cardium, right? We learn about visceral, parietal and all of this. Pericardium has pericardial fluid which is going to act as a shock absorber. Okay, which is going to act like a shock absorber. So let's say if we draw the heart like this, okay, a structure like this. If we draw heart like this, it will have a covering. Okay, it will have a covering which is called the pericardium. In between this pericardium, there is going to be pericardial fluid. Okay, pericardial fluid. What is it going to do, students? Act as a shock absorber. Shock absorber. Done? This is the general things. Okay, general things that you have to know about the heart. Yes, size is of a clenched fist. Okay, size is of a clenched fist. Okay, clenched fist. Again, men and women will have different, okay, 280 to 340 and 280, right? The different weight. Okay, correct? Yes, 12 centimeter. Very good. Okay, now moving forward, two types of circulation is there. What are they? Closed circulation, open circulation. Now, closed circulation, this is your closed, this is open. Open circulation, closed circulation, we have studied in animal kingdom. It is nothing difficult, ma'am. What is there in the students? This is your open circulatory system. Now, why do I say something is open? Okay, open circulatory system. Now, this open circulatory system, what is exactly happening? Blood is flowing through spaces, okay, spaces or body cavities called sinuses, okay, sinuses. That means here what happens, students? There is no network of capillaries, okay, there is no network of capillaries, okay, understood? Correct? No. What is endonucleus asking? Okay, body cavities, cavities called as sinuses. So, as you can see here, this is the heart, blood is coming. Can you see there are spaces, right? There are spaces. Got it? Perfect. Good job. Now, here students, we have a closed circulatory system. Okay, we have a closed circulatory system. Here we have blood is going to flow through the capillaries. Okay, here remember that the organs are going to bathe. Okay, organs are going to bathe in the blood. What is the example here? Example, you can write arthropoda, mollusca. Okay, here you can write arthropoda. Right, arthropoda cockroaches, you can remember? Yes. Closed circulatory system, on the other hand, the blood is going to flow. Blood is flowing through capillaries, or you can write proper blood vessels, right? So here the example, okay, example is going to be annelida and chordata. Done? Easy, no? Very simple. You have already studied this. So, it is no difficult, right? It is easy. Open and closed circulatory system. Now, very important, vertebrata. Okay, students, different organisms have different types of circulation. Okay, we will start with two-chambered heart. Who has two-chambered heart? Your fishes have two-chambered heart. Now, when you draw two-chambered heart, you should know there are additional chambers. Okay, sorry for saying accessory. Yeah, they have additional chambers. What are the additional chambers? Who is going to answer? I have taught this already in the class, so it should be easy for you. Very good. Sinus venosus and conus arterios. Okay, sinus venosus and conus arterios. So here when we draw the fish, okay, we can draw something like this. Okay, something like this. This meaning, okay, sorry, here. Okay, this meaning what? I'm writing the accessory organs here, but these are the exact two chambers. Okay, these are the exact two chambers. Now, students, if I see how these chambers are getting connected, I will notice that these people are taking, blood from the body. This is going to be a single circulation, right? This is going to be a single circulation. So, what is going to happen? It is going to be taking the blood from the body. Now, in between, okay, in between, they are going to have gills who are going to do the oxygenation. Okay, there is going to be gills who are going to do the oxygenation. So, it's a single circulation. Single circulation. Got it? Yes. Now, here what are we going to write? Here we can write sinus venosus, which will be attached to the right atrium. Okay, which will be, here we have conus arteriosus. Okay, these are accessory chambers. I have not written the full spelling. Okay, clear? Yes, perfect. Okay, this is going to be your atrium. Sorry, not right. Okay, just write atrium and ventricles. This is how circulation happens. Now tell me how circulation will happen. Let's say I collected all the deoxygenated blood. Okay, I'm drawing it in blue and it is going. Okay, heart is going to pump it. And where is it going to go? It is going to go to the gills. Right, gills is going to purify it. Yes, ma'am, gills is going to purify it. Okay, now from the gills, the oxygenated blood will go to the body parts. Simple. Yes or no? Any doubts in this? So here oxygenation will happen. If you see, it is just one single circulation, right? One single circulation. This is what happens in your fish. Single circulation, two-chambered heart is seen in fishes. Okay, they will have accessory organs. If you want to write it, you can write it. Okay, accessory organs. Now, when we go to amphibia, okay, when we go to amphibia, what is the scenario here? Ma'am, amphibia we have studied in frog, right? Here the scenario is, students, okay, here the scenario is they have three-chambered except for crocodile. Okay, crocodile again, amphibians, reptiles, when we talk about crocodile is going to be the exceptions. So here we will have right atrium, here we will have left atrium, but here we will only have ventricles. We will only have ventricles. Here also circulation is going to happen, but what is going to happen here? Here mixing up of blood will happen. So here deoxygenated will come, here oxygenated will come, but here what is going to happen? Here mixing up will happen. Okay, here mixing up will happen. Got it? Perfectly clear? Yes, very good. Incomplete circulation. Here it is going to be incomplete circulation. This is seen in most of the amphibians and reptiles. Amphibians and reptiles. And then comes your four-chambered heart. Okay, then comes your four-chambered heart. No doubts, I hope. Four-chambered heart, what is going to happen? Ma'am, properly four chambers are going to be there. Right, four chambers are going to be there. That's it. Everything properly, purification will happen. We're not supposed to draw it equally, okay, because your ventricles are larger. So again, right atrium, left, okay, left atrium, right ventricle, left ventricle. Okay, here proper purification is going to happen. Deoxygenated blood will come here, oxygenated blood will come here. To carry that, you have specialized systems as well. Okay, this is going to be a complete, very good, complete double circulation. Complete double circulation. Good job. Okay, good job. This is seen in your mammals, right? Crocodiles and your apes. Okay, this is done. Okay. Now moving forward, heart. Okay, students, first of all about heart, what you have to know, that heart is divided, right? Four chambers are there. For that proper divisions are there. Those divisions can be called as septum. Okay, these divisions can be called as septum. Between both the atria, that is the right atria and the left atria, you have interatrial septum, which is thin and muscular. Then for the ventricles, since more blood is going to come into the ventricles, you will need a thicker one. Okay, so you will have interventricular septum, which is thick and muscular. Now students, between this, okay, to split this two, that is the atriums and the ventricles, you have interatrioventricular septum, which is going to be fibrous. Okay, which is going to be fibrous, thin, comparatively thin. Yes, yes, we will come to pulmonary systemic circulation. Just understand the septums, okay? You have atrioventricular septum between both the atrias and ventricles, interatrial septum between both atria, interventricular septum between both ventricles. Now the question can come, which is the thicker, okay, thicker septum. All of you tell me in the chat box, which is the thicker septum? Yes. Yes, this is going to be interventricular septum. Now, can we study the heart? Okay, now we are going to draw the heart and learn the structure. Okay, learn the structure. So, until I draw, I want all of you also to draw. Okay, so that we will be able to understand the diagram well. Okay. Okay, this will take some time, students. Remember that it's heart, so we cannot draw it like, oh, one shot. Okay, fine. Okay, does not look good. So sorry. Yes, better no? Okay, let me draw the septums. Okay, and let me draw the thickenings. So ventricles are going to be more thick. Okay, the walls are going to be more thick because here more blood is going to be pumped, right? Yes. So here I'm just drawing the thickenings. Okay, I'm drawing the thickenings. Now when I draw the septum, okay, if I have to draw the septum, okay, this is how I'll draw the septum, right? Septum also done. Fine. Yes. Now look here, listen to me carefully. We are going to study the circulation. So I want all of you to pay attention for next 5-10 minutes. We will finish the structure, internal structure of the heart. It's very easy, nothing to worry about. Okay, it's very, very easy. Okay. Now students, lot of things I'll be erasing as and when I go forward, but we will study what is exactly happening. Now all of you know that oxygenated blood, okay, and deoxygenated blood is completely separate in our hearts, right? Now this is your right atrium, right ventricle, left atrium, left ventricle. No doubts in this also, I hope. Now students, all your deoxygenated blood is going to come with the help of your veins, okay? Veins, which are the two people who are going to bring all of it? I'm just drawing, okay? One is going to be called your superior vena cava. One is going to be called your inferior vena cava. They are going to open where? Into the right atrium. Okay? They are going to open into the, not now. Yeah, they are going to open into the right atrium. Yeah. They are going to open into the right atrium. Correct? Yes, ma'am, definitely. Yes. What are they called as? Superior and inferior vena cava. So I'm just writing it here. Superior. You can also call it as anterior. Okay, superior vena cava. And this is the inferior vena cava. So superior vena cava is going to bring the blood from the upper part of your body, right? Your head, neck, all these portions. Your superior vena cava is going to bring the blood. Inferior vena cava is going to bring from the lower part of the body. Correct? No doubts in this. I hope this is clear. Yes, some of you are answering, some of you are sitting ideally. I don't want any of you to sit ideally, because students, if you sit ideally, you're not going to understand anything. You have to keep answering. Okay? Okay, very good. Now students, since there is high chance for the blood to backflow into the inferior vena cava because it is placed like this, right? If something is placed like this and something is placed downwards, there is chance that the blood can come like this. So to cover this, there is valves. Okay, to cover this, there is valve which is called the valve of Eustacia. Okay, valve of Eustacia. What is this valve called as? Valve of Eustacia. I'm just telling this for your information because you need to know this. Okay, you need to know this. Okay, I think I will write it a little that side. Students, this is not for beautification, otherwise you will get confused. Okay, if I write everything together, because this notes will be available to you. So I don't want you people to make new notes. Valves of eustatia. Okay, just pray and hope nothing goes missing. Okay, done. Okay, ma'am, deoxygenated blood is now coming to the right atrium. Now, students, between the right atrium and the right ventricle, you are going to learn the first valve ever that is going to be present, which is called your tricuspid valve. Why is it called tricuspid? Because there are 3 cusps in this, okay? There are 3 cusps in this, correct? That is why you call it the tricuspid valve. So, let's say I'm drawing a valve like this, circular, okay? One cusp like this, one cusp like this, okay? So, this is your tricuspid valve, which is going to be between where? Between your right atrium and the right ventricle. Okay, fine. Okay, so we are going to label this as well. This is going to be your tricuspid valve. Also, we can call it as the AV valve. Okay, AV valve. Right, AV valve. Tricuspid or AV valve. Done? Fine. Now, obviously, if your valve is there, because why is this valve there? So that there is no mixing up of blood, at the same time there is no backflow of blood. Then here also it should be there. Yes, ma'am, definitely here also it will be there. What is this valve called as, students? It is called as bicuspid valve. Okay, it is called as bicuspid valve. Here only 2 cusps is there. Okay, here only 2 cusps is there. So this will be called as bicuspid valve. This has another term called as the mitral valve. Okay, mitral valve. This has only 2 cusps. Okay, this has only 2 cusps. Fine, done. Now, students, if you see your heart, there are going to be muscles, okay, in this lower portion. There are going to be muscles in this lower portion, okay, which are irregular, obviously not regular. Okay, they're going to be muscles like this. These are called as the papillary muscles. The larger wave, no, will be called as the papillary muscles. This one, these are going to be called as your papillary muscles. This papillary muscles is important for you to remember because your chordae tendineae is going to be attached from the papillary muscle. Papillary muscles. Done? Clear? These are your papillary muscles. Now, why did you talk about papillary muscles, ma'am? It is because, students, remember, your papillary muscles are going to help, okay, extend to the valves, okay, extend to the valves in such a way that they form chordae tendineae, okay, they form chordae tendineae, which are going to make sure that the valves are not closing and opening of the valves are regulated with the help of who? Chordae tendineae. Like this, okay, stretches like this called as chordae tendineae. So, I am just labeling that as well. These are called as chordae tendineae. Okay, tendineae, which are going to help in the closing or regulate. I can use the word regulate. Okay, regulate the closing and opening of the valves. Of valves. Clear? Done? Perfect? Yes, very good, very good. Okay, very nice. Okay, chordae tendineae. Fine. Now, students, important things. Okay, all this is important as well. Now, students, there is a small structure here. Okay, there is a small structure here which is called the coronary sinus. Okay, what is it called? Coronary sinuses. Now, this coronary sinuses is going to give all the deoxygenated blood, because heart is also, you know, muscular. That means heart is also an organ. Heart also requires blood. So that blood is going to be pumped into the right atrium for purification by the help of coronary sinuses. Okay, coronary sinuses. Coronary sinuses. Got it? This is going to pump the blood to the right atrium. Okay, right atrium. Is that visible? Yes or no? Very good. Coronary sinuses. Even this coronary sinuses also has valves which are called valves of Thesbacia. I have taught you before this. So I will just write it here. They are called as valves of Thesbacia. Okay. These are also going to prevent the entry of blood, that is backflow of blood. Okay, now students, important things. Who is going to take the blood from where? Now right atrium is going to push the blood to the right ventricle, right? Obviously ventricles are going to take it after the atrial systole. They contract, the blood is going to come to the ventricles. Now from the ventricles, who are going to take the blood? Okay, very good, Madhumati. Okay, who are going to take the blood, students? First from the left ventricle. All of you, sorry, from the right ventricle. Who is going to take the blood to the lungs? In the chat box, let's see how many of you knows this. Okay, how many of you knows this? Fast, fast, fast. Answers should come very, very fast. Some veins are there. Okay, I have to teach you one more thing. Very good, Naveen. Pulmonary artery, the only artery that carries deoxygenated blood. But how are they going to take it? So I'm going to draw all the deoxygenated, all of that in blue. Okay, blue. Yes, this blue is good, or this blue is good. Yeah. So from the right ventricle, the blood is going to be taken, okay, with the help of pulmonary artery. Okay, with the help of pulmonary artery. 1 second. Pulmonary artery is going to take the blood. No doubts in this, I hope. Okay, pulmonary artery is going to take the blood. Now students, remember this. Pulmonary artery also has valves which are called as the pulmonary semilunar valves. What are they called as? Pulmonary semilunar valves. Pulmonary semilunar valves. Got it? Pulmonary semilunar valves. Why? We have to regulate the closing and opening. Okay. Now, the purified blood, that is the oxygenated blood, will come to the left ventricles. Who is going to take this up? Aorta. Who is going to take this up? I will just draw it like this just for now, but you should understand it goes behind. Okay. Aorta is going to take the blood. You will have like this 3. Okay, aorta is going to take the blood. Oh no. Aorta is going to take the blood from the left ventricle. Okay, from the left ventricle. Any doubts in this? I will teach you pulmonary circulation and everything. Don't worry. Just focus on the diagram. Okay, diagram is important for you. That is why. Okay, they can give you the diagram and ask you to label any of this. So aorta is going to take the oxygenated blood. Okay, oxygenated blood from the left ventricles towards the body. Okay, towards the body. Now remember this, students, again. Aorta also will have valves. Okay, aorta also will have valves which are called the aortic semilunar valves. Okay, what will it be called? Aortic, okay, semilunar valves. If you learn this early, lub and dub will be easy for you. Okay, lub and dub will be easy for you. Any doubts in this? Please tell me in the chat box, and those who haven't liked the video, quickly like the video. Okay, done? Now remember this one thing. Okay, pulmonary artery. Here the openings will come here. Now I have drawn pulmonary artery to take it. Pulmonary vein. Okay, pulmonary vein openings will come here, right? Like it's, students, is actually behind, so I cannot draw it correctly. okay, the only vein that will carry, now I'll just draw the openings like this, okay. The only vein that is going to carry bring the oxygenated blood, okay. The only vein that will bring the oxygenated blood. Pulmonary vein, this is PA, pulmonary artery, okay, pulmonary artery, and this is going to be aorta. Aorta. Any doubts so far? Any doubts? Nothing, right? This is the apex, okay. This region is the apex. This region is the apex, that will be the base. So this is your whole thing. Now students, here there is a structure called fossa ovalis, okay. This is actually a small region of the foramen, but it is not mentioned, so that's why I did not teach you this time, okay. Yes. Yes, 4 pulmonary veins are there. That's why I told you 2 of them will come like this, which I cannot show. Okay, 2 of them will come like this, 2 of them will come like this. So here also like this 2 circles will be there, which are the pulmonary veins opening, okay, which will come from the down portion, which I cannot show. Okay, right? Perfect. Shall we do a revision of this? All of you, put a lot of fire emojis in the chat box so I get some energy, and then we will do a quick revision of this heart. Come on. Yes, foramen ovalis, we tell, okay, fossa ovalis, which is a small opening. Actually, it should have got closed, okay. For some kids, we tell there is a hole in the heart. It's actually the fossa ovalis which has not completely closed, okay. That means the foramen ovalis have not closed. Yes, oh, very good, very good. Some energy I can see. Okay, we need more energy. Then only we can go forward. Okay, continue. Okay, good job. Okay, let's do a quick revision. Okay, quick revision. Now students, here I have not written right atrium, right ventricle, nothing. So if there is any student who is getting confused, right atrium, okay, right ventricle, left atrium. Okay, this is how your heart is there. Okay, it is a more complex structure. I have only drawn what is necessary for you to continue. Now, moving forward, let's start from the first. First, what we have to do, we have to get the deoxygenated blood come to the right atrium. For that, we have superior vena cava, inferior vena cava, who is going to give the blood from the upper part of the body and from the lower part of the body to the right atrium. Okay. Now, in the inferior vena cava, there is chance that the blood will go inside. Okay, that is backflow, or it will, to prevent the backflow of blood, we have in the inferior vena cava a valve which is called the valve of Eustacia. If possible, remember, just keep this in mind. Okay. Now, students, there is one structure here which is called as coronary sinuses, which is going to drain the blood to the right atrium that heart has used, because heart is also an organ, right? Myogenic it is, heart is also an organ. Now, heart is myogenic, I told separately, okay. Now, between the right atrium and the right ventricle, there is going to be one valve which is called the tricuspid or the AV valve. Why is it called tricuspid? Because 3 cusp is going to be there, okay. 3 cusp is going to be there. Now, that is going to be tricuspid or the AV valve. Now, when you come this side, obviously even these 2 will have a valve. Left atrium and the left ventricle will have a valve which is called as the bicuspid or the mitral valve. Now, when you see downwards, you will see a lot of muscle, okay, muscles which are there, which are called as your papillary muscles, okay. These big rows are called as your papillary muscles. This papillary muscles actually helps like a stretch of muscle called as chordae tendineae to attach to the valves. Ma'am, why is this there? So that we can regulate the opening and the closing of the valve. Because students, they are continuously pumping, they are continuously working. We need someone who is going to regulate. So this chordae tendineae will do that job. Okay, chordae tendineae will do that job of regulating the closing and opening of the valve, which I have written it here. Done? Now, most important things, who is going to take the blood from individual ventricles, right? Now in the right ventricles, deoxygenated blood that has come from the superior vena cava, inferior vena cava, coronary veins, coronary sinuses is there in the right atrium. Now from the right atrium, atrial systole will happen, contraction will happen, blood will go to the right ventricle. Yes ma'am, it will go to the right ventricle. Now from the right ventricle, your pulmonary artery will take the deoxygenated blood. The only artery that will carry deoxygenated blood. It will take it to the lungs for oxygenation. Now after oxygenation, the blood will be taken up by the pulmonary vein, the only vein that will carry the oxygenated blood. 4 pulmonary veins, okay. Here also 2 will be there. Here also 2. The pulmonary veins will drain all the oxygenated blood to the left atrium. Now left atrial systole will happen. As the systole happens, this valves will open and the blood will flow to the left ventricles. From the left ventricles, the main aorta will take the blood. Now students, remember, aorta is not going to go and distribute the blood to everyone. Aorta is going to assign people, like renal artery, hepatic artery, like individual organs will have arteries, right? To them, this fellow will give, he telling that, okay, now this is your oxygenated blood, you supply to all of them. So like that, aorta will carry the blood from the left ventricles and supply to all the body parts. Okay, thank you so much, nitrosonium 47 AB. Perfect. Any doubts in the diagram? Now, one more thing that will be relating to your cardiac cycle is going to be pulmonary artery has valves which are called as semilunar valves. Aortic also, that is aorta also has valves called as semilunar valves. The names are very simple. Pulmonary semilunar valves and aortic semilunar valves. Okay? Yes. Small things you can learn out of NCERT. Now, coronary sinuses, it will not harm you. But you don't have to learn the whole thing out of NCERT. This is what is given in your NCERT. Now, students, in your NCERT, few more additional things are given. I mean, not additional. In this diagram itself, they have given here your SAN. Sinoatrial node, which is your pacemaker. Okay, and here they have given AVN. Okay, and then here they have given bundle office. Now I have not drawn this because it will become too complicated. Diagram already looks complicated. I do not want to complicate it more. Okay, so that is why I am not writing it. Okay, I am not writing it. I just wish to erase this septum here. Okay, so you people can see the aorta very clearly. Okay, let's just try this. 1 second. Can I make the eraser small? No. Okay, aorta. Did we erase something else? No, right? Better students, aorta, I had just erased the septum, that's it. So that you know septum does not come in between these people. So I'll just draw the septum like that. Okay. Yes. Fine. Perfect. You can make like, you know, what is that? Running notes. Okay. Okay. Rupa Shri, I am writing the notes, dear. After my class, you will get the notes. Okay. Now I am writing. No, how can I share the notes now with you? After I finish the notes, link will be made and we will share it with you. Okay. Fine. Yeah. Telegram channel also notes will be available on Biology by Gopika as well as on Vedantu NEET English. Now students, let's see the diagram. Okay. If all of you understood heart, put a lot of heart emoji in the chat box. Let me see. Will you be able to see the image that I drew? Like, will you be able to look at this image and understand? Sorry. Look at this image and tell that, okay, I have understood this much. Yes or no? It looks complicated, but it's not this complicated, right? Now, see, septums I have drawn separately for you. Now, here I have drawn all that is required for you. Okay, students, actually, No problem, Rupashri, no need to ask, sorry. Okay, so when we learn, no, in masters and all, they actually write the whole board of labelings. Full board will be full of labelings, okay, and we have to copy that. We don't have a choice. So now I have made it less for you because I know you people will feel tensed. But still, I know it will look complex, but we cannot do anything. Okay, we cannot do anything. Yes, very good. All of you understood? Pakka? Very good. Finding finding Nemo types, finding heart. Done, done, done. Okay, very good. Very good. Pakka? Perfect. Okay, now students, this is the diagram given in NCERT. Shall we go through this? Okay, ma'am, I already know vena cavas. Now, sinoatrial node, I will teach you in pacemaker. Okay, my order is little different, so we will just keep it on hold. Okay, right atrium, you know, atrioventricular node, that is again AVN node we will come into. Okay, caudate tendineae, you know, right ventricle, you know, superior, inferior vena cava, you can see aorta, pulmonary artery, pulmonary veins, left atrium, left ventricle, interventricular septum, apex. Any doubt in this? All the labelings I have taught you, some additional labelings also I have taught you. So you will have, even if they give you a diagram and ask you something, you will not have any confusion. Okay, you will not have any confusion. Students, this is your caudate tendineae. Can you see? They are like those elastic thread-like structure, no? They will make sure to help in the closing and opening of the valves. Now students, this is your cusp. Can you see? This is tricuspid. Can you see one here, and one here? Okay, like Ben's symbol. Okay, so like this. Again here, see? Can you see how they are? So these are three cusp. Okay, three cusp. Fine? Good job. Okay. Now, here you have the entire thing that you have to study. Okay, we will just go through fast with this also. See, from the tissues, we have collected the deoxygenated blood, and with the help of coronary sinuses and the great veins, that is the inferior and superior vena cava, we are giving it to the right atrium. From the right atrium, between the right atrium and the right ventricle, you have the tricuspid or the AV valve. The blood has now gone to the right ventricle. From the right ventricle, pulmonary artery will take it to the lungs for oxygenation. From the lungs, your pulmonary vein is going to give the blood to whom? To the left atrium. Left atrium and left ventricle, there is going to be bicuspal valve or the mitral valve. Okay, mitral valve. Very nice. Target completed. Perfect. Okay. Good job. Good job. Okay. Now students, from the left ventricle, the blood will be taken with the help of aorta to all the tissues. Any doubt in this? Okay. Any doubt in this? Okay. Now same thing here it is written. Okay. This I actually got inspired from one teacher, and I have done it because it's very beautiful. Okay, I don't think anyone, like, it's so beautiful, so that's why. So look here, this is the heart, lungs to the heart, oxygenated blood, obviously lungs to the heart, oxygenated blood with the help of pulmonary vein. Heart to the lungs, deoxygenated blood, pulmonary artery. Anything involving lungs will be called as pulmonary circulation. Okay, pulmonary circulation. Clear? Yes or no? Okay, now next one. Tissue to heart, deoxygenated. Heart to tissue, oxygenated. Vena cavas, aorta, systemic circulation. So from tissue to tissue, it's systemic circulation. From lungs to lungs, it is pulmonary circulation. Correct? Any doubts in this? So look here, this is also a similar flow chart. So students, as I told you, lungs is there, heart is there. So, heart will give deoxygenated blood to the lungs. Correct? With the help of whom? With the help of pulmonary aorta. Now, lungs will give heart oxygenated blood. With the help of whom? Pulmonary vein. This is your systemic circulation. This is your systemic circulation. Correct? Sorry, pulmonary circulation. Wow. This is your pulmonary circulation. Lungs to lungs, pulmonary. Okay, simple. Now, systemic means what? Tissues are there. Heart is there. Tissues will give heart what? Ma'am, tissues will give heart deoxygenated blood. You replace lungs with tissues and here again tissues and just change the names there, okay? Again heart, here. Tissues will give heart what? Deoxygenated. With the help of whom? With the great veins as well as your coronary sinuses that you don't have to write. You can write the great veins, that is your vena cavas. Okay? Heart will give tissues what? Oxygenated with the help of aorta. This is going to be systemic. So, systemic involves tissues. Systemic involves tissues. Pulmonary involves lungs. Clear or not? I have given the notes there. This I have just written to make you understand. Now, all of you answer me this one question. All of you answer me this one question. Ready? Ready to answer? Yes. Thank you so much, Rani. Yes, good job, Saubarnika. Yes. Students, who is going to take the blood from the left ventricles? Tell me, who is going to take the blood from the left ventricles? First question. First question, quickly answer. Who is going to take the blood from the left ventricles? It should be like even at night. Okay, 3 o'clock I come and I wake you up and I ask you the veins and the arteries, you should be able to answer it. That is how thorough you should be. Okay, if you have attended this class, you should be pakka thorough. Yes, so now we are going to learn cardiac cycle. Okay, we are going to learn what? Cardiac cycle. Now students, cardiac cycle when we talk about, okay, cardiac cycle we talk about in 1 minute. Okay, so 60 seconds we do around 72 beats. That means around 0.8 seconds. This 0.8 seconds is divided in the heart. Okay, very good, very good. It's divided in the heart. Okay, the circulations. Okay, circulations are divided in such a way. Just give me a minute. It's divided in what way? Both the colors are same. Okay, it's divided in such a way that 1% goes for 0.1% goes for 0.1% goes for AS, that is atrial systole. Okay, 0.3% goes for ventricular systole and 0.4% goes for joint diastole. This is what happens, right, in cardiac cycle? Yes or no? Very good. 0.8 seconds. So students, atrial systole means what? Whenever the word systole comes, whenever the word systole comes, you're going to think about what? You're going to think about contraction. Okay, contraction. Contraction. Okay, now let's draw a very okay, this is our heart. Okay, one heart, two heart. Okay, this is our second heart. Now what is going to happen, students? In 0.1 second, okay, in 0.1 second, the blood, okay, from the atria, that is atrial systole, will happen, leading to, I'm filling with random color, okay, leading to ventricles, leading to ventricles getting full, right? When atrial systole means what? The blood will go automatically to the ventricles. Blood will go automatically to the ventricles. Correct? Yes. Diastole means relaxation. Good job. Now, once the atria contract, ventricles will be full. Then the ventricles will contract, leading to the blood. Okay, now say ventricles are full. Now, because the ventricles are full, what is going to happen? It will force one to go to the pulmonary artery, right? It will tell the pulmonary artery, now you can take the blood. Okay, and one will tell aorta. Now I have not drawn the septums or anything, I'm just telling you what is going to happen. One will tell the aorta, you can take the blood, because ventricles have got contracted. Ventricles contract mean both the blood will start moving out of your pulmonary artery and that is your aorta, right? Now students, this will lead to the closing and opening of the valves, causing the lub and dub sound. Is this clear to all of you? Yes or no? Please tell me in the chat box. Is this clear? Diastole is relaxation. Yes, correct. Correct. Done? Now, oh, this arrow marks won't go. 1 second, students. Just give me 1 minute. Yes. Okay. Now, listen here carefully. Okay. So, as I told you, first AS is going, first atrial systole is going to happen. Okay. Atrial systole is going to happen. This atrial systole will lead to what? Obviously, the atria to contract. Atria will contract. If atria contract, what will happen? Yes, I am explaining again. Listen to me carefully. Atrial systole is going to happen in cardiac cycle 0.1 second. Okay, 0.1 second atrial contraction. Okay, atria will contract. Now, students, if the atria contract, automatically AV valve will be forced to open. Why? Because the blood has to go to the ventricles. So here, second thing that is going to happen is AV valve. Okay, sorry. Okay, AV valve will open. AV valve will open. So far no doubts, right? AV valve will open, correct? Superior and inferior vena cavas will close because more blood will not come. Okay, this is what has happened during the atrial systole. Next is ventricular systole. This is going to be around 0.3 seconds. 0.3 seconds. Now tell me what will happen here. What will happen here? Both the ventricles will contract. Ventricles will, okay. So this 30%, oh God. Okay. Is it fine? 1 second, let me check. Okay, it's fine, right? Okay, students, what I was telling, see, when the atrial systole happens, see, remember this way, okay, when atrial systole happens, atrias are contracting, blood will have to go to the ventricles. During this time, only 30%, okay, only 30% ventricles filling will happen. Understood? Only 30% ventricles will get full. Perfect? Okay. Now here ventricles will contract. This will lead to a isovolumetric. Now what is isovolumetric? That means here filling has happened. They have reached a situation that it's going to, like, you know, open up, right? All the valves are closed. No way the ventricles are able to, you know, hold. Like, imagine I'm closing you in a room with no windows, nothing is there. Isovolumetric contraction will happen, okay? Because all the valves are closed. Now, during this situation, what did I tell you? Now it will be forcing the pulmonary artery and the aorta to open the valve so that the blood can go outside, because it's not able to bear. No, I've closed you in a room with all the windows closed. Now you will be forced, you will try to even find a small bit where this blood can go, right? I mean, where you can go. Same thing with the ventricles also will do. Okay, same thing the ventricles also will do. So far good? So far good? Thank you, Pooja. Yes or no? Good job. So students, because I told you, they are so much in rush, they want something to open. So what will open here? Semilunar valves will open. Both the semilunar valves, okay? What are both the semilunar valves? I told you, pulmonary artery and aorta. Semilunar valves will open. So far good? Yes or no? Yes. Okay. Good job. Good job. Now, last thing that is going to happen is joint diastole. Both are going to go for relaxation. Okay. So, this will lead to joint diastole. This will take how much seconds? 0.4 seconds. This is for a longer duration, right? This is for a longer duration. Joint diastole, students, first thing remember, 70% of ventricles filling will happen during the joint diastole. 70% ventricle filling will happen during the joint diastole. Yes, no doubts in this, I hope. Yeah, we are wearing the same glasses, huh? I didn't know that. Okay, good job. Good job, okay? Students, once the blood, okay, is coming down, okay, once the blood is come down to the ventricles, AV valve will close. Why? Because there should not be any backflow of blood. That will lead to the first sound called as the lub sound. Lub sound, okay? Now, here what is going to happen? Now, the semilunar valves, here, after this, the semilunar valves, after they open, they have to close, no? Semilunar valves closing will lead to the dub sound. Okay, before the joint diastole, semilunar valves will close, leading to the dub sound, which I will write, make you understand, okay? Make you understand. Call me. Why? Why? Did you people ask about me? Okay, 70% filling will happen. Full relaxation will happen. Done? This is your cardiac cycle. This is all about your cardiac cycle. It is not complicated. You people make it complicated. It is not at all complicated, okay? It's not at all complicated. Now, students, remember one thing. All this regulation is going to happen with your sinoatrial node, AV node, bundle office and Purkinje fibers, okay? Purkinje fibers, okay? Clear or not? Some, in a circle, you will always see the cardiac cycle, okay? Now, I will teach you here. Look here. All of you look here. Yes. Listen carefully. Whatever we studied, we are going to revise once. First, atrial systole will happen for 0.1 second, right? After atrius contract, okay, after the atrius contract, what will happen? Atrial diastole and beginning of ventricular systole. That means your first heart sound will happen, because remember this way, that this is your heart. This is your heart, correct? Yes, ma'am, this is my heart. Let's take only one side, okay, let's take only one side. Let's say, students, blood was here, okay, blood was here. Now, what will the blood try to do? It will try to come to the ventricles, right? When it is going to come to the ventricles, AV valve will open. Okay, AV valve will open. Telling that, okay, blood, you go. After the job is done, immediately the AV valve will close. So, the first sound is going to be created by the closing of AV valve, which is your lub sound. Lub, okay? First sound is going to be lub. This is going to be low pitched, long duration. Okay, L for low pitched long duration. I'll give you a trick to remember this also. Now you just follow what I'm saying. Closing of AV valve will lead to the formation of lub sound. Correct? Good job. Okay, good job. I've told last time I taught you, lazy aunt will dance on Sunday. I'll make another one also and give you. Now, second thing happened. Okay, ma'am. Third thing, complete ventricular systoles. Can you see? Ventricles have got contracted. Now, ventricles have contracted means that it is forcing the pulmonary artery and the aorta to open the semilunar valve so the blood can go in, because isovolumetric contraction, full, ventricles are full. Okay, ventricles are like, okay, 30% full, now I need to push this out. So, what will happen here? Beginning of the ventricular diastole and the second heart sound will happen. That is, semilunar valves opened here. Here it will lead to the closing. So, here semilunar valves closes, leading to dub sound. This is a high pitched but short duration sound. High pitched but short duration. This is low pitched long duration. Okay, low pitched long duration. Now students, at the ending what is going to happen? Complete ventricular diastole is going to happen. Full relaxation is going to happen. Okay, during this time only your 70% ventricle filling will happen. Any doubts? Any doubts? Now you should tell. Okay, now you should tell. See here, first sound will come. Okay, first sound will come. Here what had happened? AV valves had opened. Okay, AV valve had opened. Now here closing will happen. Okay, here closing will happen, leading to the lub sound. Now what is the trick to remember this? Trick to remember this. Give me a name from A. Give me a name from A. My next class will be on Saturday. No names from A. My God, I need to bring some students who have name A. Okay, let's take A. Say what name will come? Ammu, ma'am. No, so A for Ammu. Okay, Ammu wants, new spelling. Okay, my dictionary test. Ammu wants laser bulb. Okay, I want laser bulb. Now, what is the trick for? AV valves causes lub sound. Okay, AV valve closing. Students, always remember, closing only makes sound, opening will not make sound. Okay, AV closing, that is AV valve closing. AV valve closing will lead to lub sound. Okay, lub, which is low pitched but longer duration. Okay, LL, long duration. Okay, on your NEET, May 4th, you will be able to listen. Okay, May 4th, you will be able to listen. Aslam. Too difficult to remember, no student. Arjun Reddy, what? Okay, so Ammu wants laser bulb. AV valve closing will lead to lub sound. Okay, done. Now, next one. Okay, next one. Let's say somewhere here. Okay, or here also we can put here. Okay, joint diastole, this much place if you give. Second sound will be formed here. So, this sound. So, here this sound is going to be caused by dub. Okay, that is semilunar valves. Semilunar valves closing will lead to dub sound. Okay, semilunar valves closing will lead to dub sound. Correct? This is going to be high pitched but short duration. High pitched, short duration. Done. Done. Okay. Wow. Wow, wow, wow. Excretion. Next chapter is going to be excretion puja. Okay. Yeah. Clear? So, dub sound. Dub sound is going to be heard when the semilunar valves are going to close. Okay. Semilunar valves are going to close. I had made one more trick, students. I am one, trick mafia. I am full tricks only I make. Aunt. Okay, wait. Lazy aunt. Okay, one more trick was lazy aunt dance on Sunday. One you remember. Ammu wants a laser bulb. If you remember, the next one automatically you will remember. Okay. L for lub. A for AV valve. A for AV valve. Okay. Then D for dub. S for semilunar valves. Ashte. Yes, thank you, Mohammed. Trick mafia. Yes. Done? Easy? Any doubts? Ask now. Don't ask after class. After class, where will I come and see? Who's the 20 people who did not like? We'll catch hold of them. Okay, everyone put done, done, done in the chat box. Come on. Done, done, done. One big cycle is over. Okay. Now what we have? Very less topics we have. All big topics are done easily. Good job. Okay. Palsam Shravani. If I call your name wrong, then you will get angry. So that's why I don't call any student's name. Okay. Thank you so much. Thank you so much. Yes. Okay. Perfect. Okay. This is done. Now students, arteries, veins I'll come into. Okay. Now this one. Okay. Who regulates the heart? Okay. I think I should make this in order. 1 second. Okay. This little order went off. Okay. Now where did it go? Okay. Who regulates the heart? I regulate your heart. No. Okay. Who regulates your heart? NEET examination. Right? No. Students, actually, regulation happens because of these people. Okay. How many times did I draw heart today? You'll put it in the comment section. Okay. The most. Okay. One normal heart. Okay. Normal heart. So here, what is going to happen? See students, on this corner, that is near your right atrium, there is going to be a set of tissues. Okay. Nodal tissue we tell, no? A group, a mass of cells together we call tissue. This nodal tissue is going to regulate the heart. Okay. So the first person who is going to be part of this. Okay. First person who is going to be part of this is what? SAN. SAN. Sinoatrial node. Who is he? She. Pacemaker. Pacemaker. Who will regulate the heartbeat? Who is the pacemaker of the heart? Okay. Pacemaker of the heart. Mohammed, for boards, you can join us on KCET channel, because tomorrow we are going to do a complete marathon of the most repeated questions of your, for your pre-boards as well as for your board examination. So you can join that channel. Okay. You can join that channel for that. Okay. Now students, Yes, stroke volume will come into dear. Wait for 1 minute. Okay, sinoatrial node understood. It's a set of nodal tissues, that is group of cells which will regulate the heart. From the sinoatrial node, okay, from the sinoatrial node, one more person very close to the AV valve will be present, which is called as your AV node. What will be it called as your AV node? AV node. Atrioventricular node also you can tell. Fine, AV node. Now, from the AV node, okay, from the AV node, near your septum, okay, near your septum, one person will be there who is called bundle of His. Who is called bundle of His. Basically, these people are continuing, okay. Sinoatrial node will give the, command, and then the AV node will take it up. Next person will take it up. Next person will take it up. So, bundle of His. From bundle of His, it extends all the way, okay, it extends all the way to fibers, okay, to fibers which are called as your Purkinje fibers, which are only present in the ventricles, okay, which are only present towards the ventricles. Correct? Students, actually this should be little more up, okay. The problem is when we draw heart diagram, okay, 1 second. This labeling is a problem. 1 second, 1 second, 1 second. I can just erase it, but I'm not doing it. I like the sound. Okay, so this is sinoatrial, okay, node, who is what? Pacemaker. Okay, pacemaker. Those who stay till the end of the class, I want you people to comment. Okay, I want to know your names. Right? Names of all of you. Okay, so almost near the AV node, we have, okay, AV valve, we have AV node. Okay, AV node. From the AV node extends your bundle of His. Okay, bundle of His. From bundle of His extends your Purkinje fibers, which are present spread throughout your ventricles. Okay, spread throughout your ventricles. Perfect. Okay, yes, I will tell you. So, this is bundle of His. Since Purkinje fibers is actually name of the person, okay, Purkinje. So, he only figured this out. So, yeah, Purkinje fibers. All these people, okay, all their functions is to regulate the heart, to make the heart beat. Okay, if heartbeat has to happen, what is their job? These people are all going to help. In that, specially, someone asked me, what is the Purkinje fibers? Purkinje fibers are going to be present only near the ventricle. So, these people are going to help in the ventricular contraction, that is diastole and relaxation. Okay, ventricular systole and diastole. Yes, very good. Sanushri has told that. Yes, these people are all going to transmit the impulse. Who is giving the impulse? SAN. So, SAN will give the impulse. AV will take it from there. AV will give it to bundle of His. AV will give it to bundle of His. Bundle of His will give it to Purkinje fibers. Purkinje fibers. These four people are going to regulate the heartbeat. Okay, regulate the heartbeat. Any doubts? Location is clear. Where they are present, you understood. These are all nodal tissues. Okay, nodal tissues. A group of tissues, right? Done? Yes. Yes, correct. Conduction of electrical impulse. Okay, they help in the conduction of electrical impulse. That is their job. Okay, then only we will be able to get the beats. Okay, regulate the beats. Done, done. Very good, very good. So, who regulates the heart is clear, no? Who regulates the heart is clear? So, here you can see the more better diagram. Okay, so here you have sinoatrial node, okay, SA node, atrioventricular node, AV node. From there you have bundle of his, can you see in the center? Then you have Purkinje fibers extending to the ventricles, which will help in the diastole and the systole on the ventricles. Okay, these people will regulate the heart. Okay, next is blood pressure, stroke volume, all of that. Okay, after this we have only ECG, and then we have about arteries and veins. Then we are done and all some of the disorders. Okay, some of the disorders. Okay, very good. Those students who stayed back fully, good job for you. Okay, I'm proud of you. Yes, blood pressure. So students, the pressure exerted by the arteries. Okay, pressure exerted on the arteries, right? Pressure exerted, yeah. What is this? Bulbulaya 4 on my board. Okay, pressure exerted on the arteries by the blood, right? Okay, arteries. Now remember this. Remember this. See, whenever, okay, whenever exerting, okay, we always tell no normal pressure. Normal pressure. Now all of you have normal pressure. When will your BP be high? When will you feel hypertension? On May 3rd. Okay, even I'll have hypertension, even you will have hypertension, right? So normal pressure is going to be how much, students? 120 by 80. 120 by 80. Now this 120 and 80 has a thing. What is this 120? It is the systolic pressure. Systolic pressure. Divided by, okay, divided by, no, like by, okay, diastolic pressure. Diastolic pressure. Correct? Correct? May 4th and May 3rd also you people will have little bit. Okay? Little bit. Okay? 120 by 80. Now what is hypertension? What is hypertension? All the letters are going. Now I'll have hypertension. Okay? Hypertension will be 140 by 90. Okay? 140 by 90. Now this can range, students. Okay? There is actually, when only, there are situations where only diastolic pressure is high. There are situations when only systolic pressure is high. This is also seen in people. Okay, it's not necessary that every time it is 140 by 90. Sometimes it can be 140 by 80. Sometimes it can be 120 by 90. There are chances that some people's only diastolic pressure is high. Some people's only systolic pressure is high. So this can range from 100 to 120. Okay, and this can range from 60 to 80. Okay, 60 to 80. This is 140 to 90. Okay, 140, yes, mm of Hg. Okay, that you can just write it. I'll write it here. Mercury. Okay, mm of mercury. Now again, this is also diastolic pressure and this is also G went somewhere. Ayyo. Okay, mm of Hg. Got it? I hope no doubts here. Very good. Okay, now we can calculate pulse pressure by minusing systolic pressure minus diastolic pressure, pulse pressure. Okay, yes, fine, this is done. Hypertension, how will we measure this? With a sphygmomanometer. Okay, with a sphygmomanometer we will measure it. All of you have seen, no, we'll tie like this, we'll press like this, and up to we reach the actual value. Okay, that's how we get to know. Now students, stroke volume. Okay, some of you are asking me about the stroke volume. No, now stroke volume, first of all you should know what is stroke volume. Stroke volume is the amount of, you know, blood. Stroke volume is the quantity or the amount of blood that is coming out of the ventricles, okay, during ventricular systole. During ventricular systole. Oh my god. Okay, this dance, students. I think I have to give 2 minutes break for the board. Board is shouting. Okay, so it is the volume of blood which is ejected out by ventricular during the ventricular systole. Okay, so ejected out. So that is usually 70 ml. You can use the measurement, okay, that is the diastole volume minus two minutes. Let the board be little calm. Okay. Okay. Yes. Very good, Tanushri. Okay. How will you calculate it? With the help of EDS, end diastole volume. Okay. ED. You can tell neither systole, or you can just write the volume as well. Okay. So end diastole volume minus end systole volume. End diastole volume, students, when the ventricles are fully filled. Okay, when the ventricles are fully filled. Okay, so that will be during the relaxed state, it will be around 130. Oh God. Okay, or else it will be end systole volume. Okay, so 130 and your EVS normal relaxed state will be 60, which will lead to 70 ml. You can just write 70 ml because your calculation no one will ask you, but I'm just telling you the calculation for those students who asked me. Okay, this is how you can calculate it. EDV minus ESV, that is diastole volume, systole volume, that is when the ventricles are full. Okay, so end diastole volume and end systole volume, 130 minus 60, which will give you 70 ml. Now 5040 also you know, no, stroke volume into the number of heartbeats. Now students, remember, you don't have to do the value until they have asked you, right? Your cardiac output, when they ask you, they will give you the number of heartbeats. What do we normally take? We take 70 into 72, which is equal to 5040, okay, ml, which is equal to 5 liters of blood. Correct? Now, sometimes, even in boards also, I have seen questions where they give the heartbeat. They tell, okay, it is 69, 54. That time, okay, that time you have to calculate it. Okay, correct? Yes, very good. Cardiac output, when you're calculating, if they give you the heartbeat, you please don't put 72. Wait for them to, like, read the question properly. Otherwise, generally your cardiac output will be stroke volume into the heartbeat. Okay, number of heartbeat, which is equal to 5040 ml. 5040 ml, approximately you will take 5 ml. That is your entire volume of blood that is entering and coming out. Perfect. Done, done, done. Okay, done, done, done. Okay, 2 seconds break. Okay, just for you to copy and for the board to relax. Fine? No doubts, no? Who asked me about, who is that? Someone asked me the question about calculation. Is it clear? Sphygmomanometer, you please write it. Okay? Very good. Good job. Okay. Calculation done. Okay. Now we have ECG, artery and vein. Okay. That's it. Then we are full done, done. Okay. We are full done, done. Okay. Good job. Now, students, nothing here. I just want you to just know the layers also. You don't have to know, but then just let's just go through this. So when you take your arteries, okay, arteries and vein, what is there? 3 layers are there, no? So the external layer will be tunica externa, middle layer will be tunica media, middle, okay, media, and the last layer will be tunica intima. Okay, these are made up of your squamous endothelium. Okay, because we have learned, no, squamous endothelium lining of the blood vessels. So these are smooth muscles and elastic fibers. Your tunica media, and your tunica externa will be fibrous connective tissue with collagen fibers. You don't have to worry about this, but worry about squamous endothelium. Okay, tunica, the last layer, intima, is made up of squamous endothelium. This you remember because it's a part of your syllabus. Okay, it's a part of your syllabus. So if you take the structure, this is what you will see. You will see an outer layer, a middle layer and an inner layer, in which the middle layer is more wider. Okay, middle layer is more wider. Now about the lumen. Artery lumen is bigger, artery lumen is smaller compared to the ventricle lumen. Lumen is a small in the between space through which the blood is going to flow. Now vasoconstriction, vasodilation is actually where the lumen will extend a little. Okay, that is done by your histamine and serotonin. No, that is where your contraction, relaxation, little movement will happen. Your veins, the blood, the flow is going to be faster and it is going to be more. So that's why the lumen is bigger here. Here the lumen is smaller, but the arteries are going to be thicker. Can you see the thickness here? Can you see the thickness here? Can you see? Because arteries have to take blood away from the heart. Okay, because arteries have to take blood away from the heart. More distance to go, right? This has to bring blood towards the heart, correct? So that's why the thickness difference is there. This is just for your understanding, okay? All of this I know you have studied in your smaller classes. Now students, ECG. ECG, now the confusion with ECG is the instrument name is electrocardiograph. Huh? Ma'am, graph?
I can't do that.
Okay, my Siri got activated. I don't know why. Okay, so the confusion here is the instrument is called as electrocardiograph. But the one that you get, okay, the graph that you get is called as electrocardiogram. Clear? Am I audible? Please type something in the chat box, otherwise I will not know when I am lagging. Okay, your chats is equal to my movement. Okay, because I can't see my live, na? Okay, perfect, perfect. Keep answering something. Okay, so students, the instrument is called what? Electrocardiograph. Okay, electrocardiograph. And the graph that you get, right, the results or the graph that you get, that is going to be called electrocardiogram. So you are studying the electrocardiogram. Okay, you are studying the electrocardiogram. Okay, yes. Students, why do we study this? So when a doctor looks at our ECG, they will get to know that do we have any issues, that is our heart functioning normally, because they will be able to see atrial systole, ventricular systole, ventricular repolarization. They will be able to see all of this. By this they will get an idea if the heart has any problem. Now in some cases we tell heart murmuring, that means the variations will be a little different. Okay, now in all of your serials, mothers will be seeing serials, no, that time what will happen? First actor, the something like this will be shown, right? After the heroine comes, okay, after the heroine comes, when the heroine is near to the hospital, the fellow's heart will become like this. Okay, his heart, his ECG will show like this. And when beep sound will come, okay, beep sound will come, your mom will start crying, heroine will start crying, everyone will start crying. But what exactly happened, students? What exactly happened? Tell me in the chats. Okay, what exactly happened to him? What exactly happened to him? His heart stopped beating. That means he is no more, right? He's no more. So that is why, that is when this will come, this beep sound, and your entire ECG coming to a blank state. Okay, that is when your mom will cry, your dad will think that something happened to you, but nothing would have happened. It is basically a serial fellow who is died. But this only happens when the heroine is very near to the hospital, or she's, you know, between two lorries, or is she in a traffic jam. That time only this will happen. Okay, otherwise it will not happen. If the heroine would have reached the hospital, then he wouldn't have died, right? So such scenarios happen. They keep using ECG without knowing which is ECG, which is electrocardiograph, and which is electrocardiogram, but you should know it. Now students, usually your electrocardiograph will be having 3 leads. Okay, 2 usually to the wrist, okay, and 1 to the anklet. Okay, 1 to the ankle, ankle, 1 to the ankle, but it can go up to 12 also. Okay, it can go up to 12 leads also. Nowadays 12 leads also is there. This will exactly tell what are we capturing on. Okay, yes, a cardiac arrest happened to him. Okay, yes. So here you can see, you know, multiple leads can happen, depends. Now you have to know what? You have to know different types of waves. that is important for you. You don't have to know anything about the serial. So here, first wave. What is this? This circle, ma'am, this circle is atrial systole, also called as what? This is where the atrias will contract. Oh, first I'll label it, sorry. First, this is P. P, Q, R, S, T, U, V, W, X, Y, Z is not there, okay? U, V, W, X, Y, Z is not there. So in the examination, don't write whatever comes in your mind. Oh, U, V will be there. Okay, this is going to be atrial systole, or else in other words, given in your NCERT, what is that? Atrial depolarization. Depolarization. Okay, depolarization. All of you know the sodium ions coming in, depolarization, repolarization. Yes. Then next, the most important wave, okay, QRS. Okay, most important wave is your QRS, because it's one of the biggest wave and you can easily spot by looking at the wave. You will easily able to know, okay, calculate the beats as well. Okay, so here you have your QRS. Now what is QRS? Very good, Roshni. Ventricular depolarization. Okay, ventricular depolarization. Yes? Okay, I had 2 minutes of confusion of the names. Got it, all of you? QRS we can calculate to know the number of beats. Okay, QRS is very important wave, one very important wave. You can also say it as the ventricular systole. Okay, good job. Next, what we have? Ma'am, you have a Kutti T wave. Okay, one Kutti T wave is given. What is this T wave for? This is going to be ventricular repolarization. Ventricular repolarization. Now you can ask me question, ma'am, what about atrial repolarization? Students, atrial repolarization also happens, but it is not big enough to wavelength is not big enough to get captured on the ECG. Okay, it is not big enough to get captured on the ECG. Got it? Yes? Yes. Tell me which is the highest amplitude one? Highest amplitude. Highest amplitude is going to be R. Yes or no? This is going to be the highest. Okay. This has the highest amplitude and S has the lowest amplitude. If you see properly, S has the lowest amplitude. I told by mistakenly wavelength, I guess. I'm sorry, amplitude. Okay, lowest amplitude. Got it? This is how you will be able to denote it. Okay, students, this will allow the doctor to know to a limit, okay, if there is a problem or not. Okay, now looking at this, the doctor will be able to know if the heart has any problem or not. So this is how your ECG will be shown. Okay, this will be how an ECG will be shown. Now during normally how do we see it? Like this, right? Like this we see it in the serials and everything, we see like this. Okay, but when you're studying you see like this, correct? Like this. So ECG, this will let us know if the heart has any issue, okay, any problem is there with the cardiac, is the person going to have myocardial ischemia, all that we will get to know, at least to a basic level we will be able to know by looking at an ECG. Any doubts in this? Easy, no? Okay, atrial repolarization also happens, but the amplitude is not big enough to get captured on an ECG. So this is atrial depolarization, P for QRS, depolarization of the ventricles, and repolarization leads to ventricular repolarization is T. Okay, T. Okay, done. Okay, now answer these questions. Yes, answer the questions. Students, I'll get some water and come. Don't go anywhere. Just answer this question. Don't. Okay, answer this question. I'll be back in a minute. I need some hot water. That's why. Yeah. Keep answering this question. I'll come. Yes. Perfect. What's the answer? D is the answer. Good job. T is the answer. So you got 12, right? No, 16. So this is going to be 20. Okay, 20. Those who got 20, don't forget to drop it in the comments. I mean, before also. Now, which one of the following statements is correct? Oh no. Students, I'm so sorry I showed the answer. Okay? But you can just go through the question. It's a 2022 question. Okay, which one of the following statements is correct? The atrioventricular node generates an action potential to stimulate atrial contraction. Tricuspid and bicuspid valves open due to the pressure exerted by the simultaneous contraction of the atria. Blood moves freely from atria to ventricles during a joint diastole. Okay, option 3 was the answer. Okay, next question. Yeah. Match the items given below, column 1 and column 2, tricuspid valve, bicuspid valve, semilunar valves. Yes, answer fast. Answer fast. Yes. Quickly. Tricuspid valve, bicuspid valve, semilunar valves, between whom and whom? Fast, fast, fast. No time to waste. Tricuspid valve is going to be between the right atrium and the right ventricle. Bicuspid valve between the left atrium, left ventricle. Semilunar valves between right ventricles and pulmonary artery. So A3, only one option is there, option number C. So plus 24 for me. Okay, I get 24 marks. You people don't get. Okay, you people are very slow. Next one. Which one of the following human organs is often called the graveyard of RBC? 2012 question. This you people have to answer fast. I think in my phone it should be faster. Yes, quickly. Let's see who's the fastest finger. Those who haven't liked now, okay, your heart will regret. So quickly like, okay. Don't make your heart regret. Very good, very good. Okay, spleen is the answer, right? Spleen is the answer, plus 28. I want all those who attended this class to put their marks in the chat box. So out of 28, I want all of you to tell me how much you got. Okay, how much you got out of, sorry, my maths teacher is going to cry seeing this. Okay, plus out of 28, how much you got? If you made one mistake, you minus 4 from it. No need minus 5, minus 4 from it. Okay, if you unattempted it, you haven't attempted it, put 0. Like, you just leave it. Okay, yes, we have reached the target. Very nice. Good job, students. Good job. Proud of you. Okay, now the diseases. After that, we are done. Okay, we have solved 28 marks questions. Okay, the chapter weightage is around 12 to 16 marks, but these kind of questions only will come. Only your concept knowledge is important. Okay, don't be like, oh my god, heart diagram, will it be so difficult? Nothing is so difficult. Pulmonary systemic circulation, you understood it very clearly. Cardiac cycle also you understood. One will open, next one will open, closing, valves will close, sounds will be made. Lup sound when AV valve close, dup sound when semilunar valves close. Easy. Okay, lazy aunt lazy aunt dance on Sundays, or Ammu wants laser bulb. Right, simple. Okay, just remember the trick. Okay, and then if you want to remember all the orders of WBCs, what is that? Never let Mohan eat bread. Neutrophils, lymphocytes, monocytes, eosinophils and basophils, depending on the order. Then you can also remember the granulocytes with the name Ben. Okay, Benton's Ben is there, no? Ben you can remember. Monocytes is LAM, L-A-M. That is L for lymphocytes, M for monocyte, A for agranulocyte. For everything, trick is there. So all you have to do is study. Now, coronary artery diseases, that is CAD disease, also known as arthrosclerosis. Okay, arthrosclerosis, caused by buildup of plaque, that is your calcium, fat, fibrous tissue, makes the lumen of arteries narrower. Students, if the lumen of the arteries is narrower, the pressure at which the blood is going to come is going to be more. Okay, that will cause a problem. Affects supply of blood to the heart muscles. Obviously, if arteries are supposed to be like this, let's say in the arteries, there is accumulation of calcium or, plaque has happened. Now what will happen? The blood will have to move through the smaller gap. That will definitely affect the amount of blood that is going to the heart. Okay, plaque is like, have you seen on your teeth also? Sometimes you get this yellow color deposition. You can call it as start, but starting it is plaque. That means accumulation of something. It could be accumulation of calcium, especially on your teeth. Okay, fluorine ion is there. So because of that, plaque formation happens. Okay, a thin layer of yellow color deposition. Okay, thin layer of yellow color deposition. Clear, Tanu? So that is your CAD. Now students, CAD can lead to everything else. Right, if the arteries lumen is narrower, it will lead to the supply of oxygen becoming less. Right, the amount of blood that is coming to the heart will be less. The amount of heart pumped by the heart will be less. So it will affect everything. Okay, it will affect everything altogether. Now, angina pectoris, also called as angina pectoris, not enough oxygen reaching the heart muscle. So here it is because of lack of oxygen. Lack of oxygen. Okay, now symptoms are acute chest pain, can occur in men as well as in women. Okay, here what is happening? Again, this is also I told you, when the plaque accumulation happens only, the amount of blood that is going to come to the heart will reduce. Automatically, it will stop supplying the amount of oxygen also. Heart is also an organ. It needs oxygen. It needs oxygenated blood. So here, when plaque accumulation happens, the amount of blood that will move. Okay, if say this much, in this much gap, let's say I can take this much chocolates. That means I'm going to take more. Let's say my one hand is not there, one hand only. My reduction, right, the amount of chocolate that I'm going to grab is going to reduce. Okay, so same thing here also. This much blood can go, okay, or this is the lumen. This much blood can go. Let's say here it is covered like this, okay, some plaque is there. Now the space is going to reduce, okay, space is going to reduce. Now, also now myocardial infarction, okay, which is called as your heart attack, no? Plaque ruptures and causes blockage. That means, let's say here your arteries are there, okay, the plaque was there, it ruptured, okay, it ruptured and let's say it went and blocked in the middle. Now what will happen? The blood vessels the blood will not be able to move. This can lead to damage or death of cardiac muscle tissues, okay, this will lead to the death or damage of cardiac muscle tissues, which will again block the supply. So students, here you can see the difference. See, coronary artery disease, plaque builds up in an artery. Again, plaque build-up increases and it is harder for the blood to get through the artery, leading to angina pectoris. Now, if the plaque ruptures, that will lead to heart attack, which is also called as myocardial infarction. Do you understand the difference? Most of the students do not understand the difference. With this image, that difference will be very beautifully clear. Here, plaque accumulation has increased, because of which the amount of oxygen that has to reach to the heart has reduced. Okay. Let's say a lot of buses are coming in your area. Suddenly buses have reduced. That means the amount at which, the speed at which, the amount at which students will go to college will automatically reduce. Now, plaque has ruptured. Okay, rupturing has happened and now it has blocked. Now the blood which is coming will start going back. The blood which is coming will start going back, leading to heart attack, because the cardiac muscles have got damaged. It will arrest, okay, that is heart attack will happen, cardiac arrest will happen, okay. Arrest is different, attack is different. I've taught you this in that question also, okay. Now, atherosclerosis and arteriosclerosis, you have to know the difference. Atherosclerosis, also known as CAD, okay, also known as CAD, is when the plaque accumulation happens, okay. But atherosclerosis is when thickening happens. See, thickening of the inner wall and the central wall of your arteries. That is when your inner wall thickens. Okay, that will lead to atherosclerosis. Okay, that is this is arthro, this is arterio. Okay, arterio. Any doubts in this? Any doubts? Please make a note. Okay. Okay, got it? Yes. Students, this will automatically understand, all these diseases will automatically affect the kidney, okay, liver. Obviously, it's going to affect everything, right? And I hope all of you know that kidney and the lower part, right, the intestine part will have, again, even your liver, intestine, they all have portal systems, right? So there is renal portal system, there is hepatic portal system. All this is going to get affected. Okay, hepatic portal vein, renal portal vein, all of this are going to get ultimately affected. All of this are going to get ultimately affected. Done. Okay, how many of you got 28 marks? Please drop it in the chat box. So students, I know all of you know about this. Okay, again, 15th Jan, that is today, the batch has started. So I want all of you to not be any more late. Join the batch right now. These are the things that you will be getting: live online test, pedagogy and practice sessions. You will also get V-Buddy, where you can get topic wise lectures. Okay, and you will not get at home online AITS, but you will get it here in this, okay, combo. That means, again, don't spend money on test paper. Just take the combo, because in this you will get access to all the ebooks, you will get access to all the tests. Every Sunday you will have mock test. So I would recommend all of you, please go for this combo, because at 3,500 you will get All India Test Series. At the same time, you will get all the recorded lectures. Whichever lecture you want JEE, you want NEET, everything will be available. Okay, and you will also get, every Sunday you will get mock test from 2 to 5. Okay, every Sunday you will have class test type, no, from 2 to 5 p.m. for the topics that has been done on that week. Plus, you will also get All India Test Series delivered at your home with the OMR sheets. Okay, with the OMR sheets. And I told you, if you don't know to mark your OMR sheets, you can even go to negative marking. So all those students who enjoyed the session, without fail, drop it in the comments how the session was. And also, please let me know your marks. Okay, how much marks you have got for the, GM question round. Okay, GM question round, where I put the questions for you. Do let me know. And students, this beautiful notes which I have written for you, you will be getting it after some time or by tomorrow morning in the description box. Okay, Thank you so much for joining. I want all of you to take that 1 minute to comment and let me know how the class was. Even if it was like, ma'am, class was good, that's more than enough. Okay, because it will give the trust for another student to open the class and see. And this is going to be a one-shot body fluids and circulation. I hope everything is super duper clear to you. Any doubts, please drop it in the comments as well, and also drop your marks. Okay, I'm excited to know those who got 28 marks out of 28, those who got 20, those who got 24 also. Good job. Thank you so much. See you all. Good night. Take care and sweet dreams.